Welcome back, and welcome to Unit 2 of our Introductory Microbiology course. In this particular slideshow, I'm going to do several things. I'm going to begin by reviewing some of the material that I talked about in the last lecture, so hopefully that won't be too boring for you. And I will also discuss it in a little bit more detail. So we're going to start out by discussing the different types of microbes that are used, or different types of microbes that are studied by microbiologists. We will then have a brief discussion about cell biology. Some of you have already taken cell biology at Columbia. That would be the Biology 200 course, and so you'll be very familiar with the material. For others of you, it will be new material. So we'll review the shape and structure of cells. We'll review what all of the little organs that are present inside the cells do. Of course, if you have organs inside a cell, we call those organelles. And then we'll move on to a review of the types of microscopes that we use to look at microorganisms. And then finally, we'll have a brief discussion about some of the stains that we use in order to see or visualize these microbes when we're looking at them under the microscope. Okay, so we'll start out with a review of the structure of different cells and different organelles. All right, so the most fundamental difference between living things on Earth is the first thing you would ask whenever you see any living thing is you would ask, is it a prokaryote or a eukaryote? In the most simple terms, the difference between a prokaryote and a eukaryote is that a eukaryote has a nucleus. A eukaryote contains its genetic material in a nucleus, in a special compartment called a nucleus. A prokaryote does not have a compartment called a nucleus. Other obvious differences between prokaryotes and eukaryotes are that prokaryotes tend to be much smaller than eukaryotes. By the way, prokaryote is just a fancy name for bacteria. So bacteria, as I've said many times before, human life on Earth, we believe that life on Earth, we believe that the Earth is about 4.5 billion years old, and that during the first 2 billion years there was actually no life on Earth at all. And then suddenly during the 3rd billion years, bacteria appeared. And so bacteria, in fact a special type of bacteria called archaebacteria, evolved, and evolved spontaneously from the primordial soup that contained nitrogen, hydrogen, oxygen, and carbon, and so on. And so bacteria evolved first. And so for 1 billion years, we had nothing but bacteria on Earth. And then for another 1 billion years, in the 2nd billion years, I guess that would be the 4th billion years of the lifespan of the Earth. In the 4th billion years, eukaryotes evolved, which were larger single-celled organisms that were larger than bacteria, and they contained their genetic material in a special compartment called the nucleus. And then in the last 500 million years, which is practically no time at all, we went from having just boring old bacteria and having boring old protists, because protists, of course, are single-celled eukaryotes. We evolved from bacteria to protists, and then from protists to everything that you see now. The Earth is now exploding with different forms of life, so many different forms of life. But that is very recent in the history of the world. So for 1 billion years, we just had prokaryotes, bacteria. Then for another billion years, we had bacteria and protists that are single-celled eukaryotes. And then for the last 500 million years, we had all of these wonderful multicellular organisms that you see on the Earth. All right, so the first question that you would ask if you see a living thing is you would ask, is it a prokaryote or a eukaryote? The main differences between prokaryotes and eukaryotes are that eukaryotes contain their genetic information in a nucleus, while prokaryotes do not have a nucleus. Another obvious difference is that eukaryotes are much larger than prokaryotes. So a typical prokaryote is around 1 micrometer in size, and a typical eukaryotic cell, a typical eukaryote cell, is maybe 10 microns in diameter. Okay, the third difference is that eukaryotic cells contain a number of different fairly large membrane-bound organelles. So you know that the human body is formed of different organs. We have a heart, we have lungs, we have liver, and so on. Those are called organs. Well, it turns out that a cell has little tiny organs that do carry out specific functions inside the cell, and we refer to those as organelles. Organelles. And the one of the differences that so one of the organelles that both prokaryotes and eukaryotes have is called a ribosome. You may recall from first year biology that a ribosome, the purpose of a ribosome is to synthesize or translate proteins. Okay, so that's what a ribosome does. But the ribosome is a fairly small organelle. It's a fairly small and simple organelle. But you know that eukaryotic cells have larger, more complicated organelles, like the mitochondria, like the endoplasmic reticulum, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Those are larger, more complicated organelles, and those organelles are surrounded by their own membranes. Right, so if you have larger organelles that are surrounded by their own membranes, that's something that only eukaryotic cells have. In fact, excuse me, in fact, the larger organelles like the mitochondria, we believe, were actually at one time bacteria that lived independently, and at some point a eukaryotic cell swallowed a bacterial cell with the intention of eating it and dissolving it and digesting it, and by accident it didn't digest it. It didn't dissolve it or break it down and eat it. It just left it alone, and that bacteria cell thrived, and it produced so much excess energy that the actual cell that had swallowed it benefited from it. So mitochondria are sort of energy producing slaves that were once independent, free living bacteria. Okay, so what that means is that a prokaryote can't have mitochondria inside it because it's not big enough. In fact, a mitochondria is exactly the same size as a bacteria, and that's not a coincidence. That's not a coincidence. They are the same size because mitochondria used to be independent, free-living bacteria. All right, now, so once you've asked whether a cell is a eukaryotic cell or a prokaryotic cell, or once you've asked, you have some newly discovered organism, the first thing that you will ask is it a eukaryote or a prokaryote? Does it have a nucleus? Are the cells big? Is the DNA kept in a separate compartment called a nucleus or not, right? So is it a eukaryote or a prokaryote? All right, the second question you will ask is, does it exist as a single cell or does it exist as a multicellular organism? So we've already learned that prokaryotes do not form multicellular organisms. They live independently. Every cell is a complete organism. Right? The same thing is true for protists. So protists are single-celled eukaryotes. Right? What about humans? Humans are multicellular eukaryotes. Each human being is made of billions of different cells, and the cells are all specialized. Right? So the humans are made of billions of cells, and a heart cell is different than a brain cell, and a brain cell is different than a kidney cell. So these cells that multicellular organisms are made of tend to be specialized and what are called differentiated. So the process of differentiation means that for a cell to go from being a generic general cell into being a specialized cell. That's called differentiation or cell differentiation. All right, so here we have a cartoon diagram of a eukaryotic cell. You can see that it has a number of large internal organelles. The nucleus itself is considered to be an organelle, and the nucleus contains the genetic material. The endoplasmic reticulum is also considered to be an organelle. The Golgi complex is considered to be an organelle. The different mitochondria, each cell has about half a dozen mitochondria. That is considered to be a different organelle. The plasma membrane is the layer of phospholipids that surrounds the cell and defines the inside of the cell versus the outside. So let's take a look at some of these organelles. All right, so the plasma membrane, beginning with the plasma membrane, and in some cases a cell wall and or a periplasm. All right, so every cell, every type of cell, whether it's a prokaryote or a eukaryote, is surrounded or the outside is defined by what's known as a phospholipid bilayer. So a phospholipid bilayer, you may recall from first year biology, it's made of phospholipids. There are two layers of phospholipids, and you may recall that phospholipids are hydrophobic on one side and hydrophilic on the other side. That means that the hydrophobic sides will get together so that they can be away from the water, and the hydrophilic sides of the phospholipids will be deliberately facing the water because they like to be dissolved in the water. So if you take phospholipids and you throw them into a jar of water and you shake it up, they will spontaneously turn into little cells. They will form a phospholipid bilayer spontaneously because that's the only way that a molecule that's hydrophobic on one side and hydrophilic on the other side, that's the only way that it can deal with being in the water. All right, now a phospholipid bilayer is actually quite flexible and fluid-like. And so we don't want to make a strong solid cell, we don't really want the phospholipid bilayer to be that flexible. We want it to be a little bit more rigid. And so the body makes cholesterol, right, and inserts cholesterol. Cholesterol is a ring structure where you take you take one of the phospholipids and instead of leaving it as a chain, you bend it into a bunch of ring structures. Those ring structures are not flexible, and so they will instead of if you insert cholesterol into the phospholipid bilayer, it will cause it to become more rigid and less flexible. Right, so you've probably heard that cholesterol is bad for you. That's actually not true. We need cholesterol in every cell in the body. We can't get by without cholesterol. What's bad for you is having too much cholesterol in your blood. And that usually happens by eating the wrong kinds of foods. But besides that, cholesterol is good for us. Not only is it good for us, but it's essential. We couldn't live without it because every cell, the plasma membrane of every cell contains cholesterol. All right, now what is a cell wall? Well, some organisms have, particularly plants, have another layer, kind of another shell outside of the plasma membrane. And that cell is made of, if it's a plant cell that we're talking about, that cell wall is made of a polysaccharide called cellulose and another polysaccharide called lignose. Right. Now, so plants are an example of an organism that has every cell is surrounded by a cell wall. Okay, so animal cells are not surrounded by a cell wall, right? They're not. So don't confuse, it's a common mistake to confuse the phospholipid bilayer with the cell wall, but they are two totally different things. And so animal cells and a lot of other organisms, the cells are not surrounded by a cell wall, but every cell has a plasma membrane. Every cell has a plasma membrane, but every cell does not have a cell wall. It depends on the organism. Okay, so plants are an example of one organism that has a cell wall, and bacteria are another example. Some bacteria, not every type of bacteria, but some specific types of bacteria are surrounded by, the bacteria has a plasma membrane like every other cell, but some bacteria, in addition to the plasma membrane, have an additional layer called a cell, which is also referred to as a cell wall. But unlike plants where the cell wall is made of cellulose and lignose, with bacteria that have a cell wall, the cell wall is made of a polysaccharide called peptidoglycan. Peptidoglycan. Memorize the word peptidoglycan because it'll probably be on a test at some point. I was forced to memorize it when I was, 20 years old. Everyone has to memorize it. There's no way around it. So some bacteria have cell walls outside their plasma membranes, and those cell walls are made of peptidoglycan. So that's something so a peptidoglycan cell wall is something that certain types of bacteria have. All right, now there are other types of bacteria that do not have a cell wall, but instead they have another plasma membrane. So in other words, they have two plasma membranes, one inside the other. Right? And when I say two membranes, I'm not referring to the phospholipid bilayer. I'm referring to two phospholipid bilayers. Right? Now, there are some bacteria that instead of having a plasma membrane and then a cell wall, instead they have a plasma membrane and then another plasma membrane. And there's a little tiny space in between those two plasma membranes that we refer to as a periplasm, or a periplasmic space. Right? So the term periplasm or periplasmic space refers to the space in between the two plasma membranes of certain types of bacteria. You can also refer to the periplasmic space as the space in between the cell plasma membrane and the cell wall, but that's less common. All right, couple of terms that we need to review. Endocytosis means bringing things into the cell. Exocytosis means sending things outside the cell. Furthermore, all cells, virtually all cells, and certainly human cells, have things on their surface that are referred to as receptors. So cells send signals to other cells, particularly in a multicellular organism, where the signals, instead of sending radio signals or television signals, they send signals in the form of little proteins called cytokines or little lipids called hormones. And in the case of protein hormones, signals, cytokines, and growth factors that are made out of protein, those signals are received by receptors that are on the surface of the cell. Right? So we have cell surface receptors that receive signals. Those signals are usually in the form of little proteins or hormones or lipid hormones. Right? Okay, and another word we need to be familiar with is if you have a phospholipid bilayer, the area inside the phospholipid bilayer is referred to as the cytoplasm. The cytoplasm. So remember that the word cyto means cell. It's an ancient Greek word meaning cell. And then plasm means, kind of a sticky stuff, sticky liquid inside called a plasm. All right, so here we have a diagram of the phospholipid bilayer. Notice that this is a phospholipid bilayer that's organized with the hydrophilic phosphate heads facing the water on the outside of the cell as well as inside the cell, and the hydrophobic tails facing each other on the inside of the phospholipid bilayer. This is an electron microscope image of the phospholipid bilayer. The scale, remember that if you take a meter stick and you divide it by 1,000,000, you have a micrometer. If you take a meter stick and you divide it into 1,000,000,000 parts, you have a nanometer. So we measure the sizes of cells in micrometers, microns, and we generally measure the sizes of many of the smaller organelles inside a cell. We measure those sizes in terms of nanometers. And it just so happens that the phospholipid bilayer that you see here in this microscope image is about 7 nanometers in thickness. Right? So the phospholipid bilayer of any cell is around 7 nanometers in thickness. Here's another diagram of the phospholipid bilayer. In this one, we can see a little bit more detail. We can see that there are many proteins that are embedded in the phospholipid bilayer. These proteins are all embedded. And here we have some cholesterol to make the phospholipid bilayer a little bit more rigid. Right? This area outside the cell is referred to as the extracellular area or the extracellular space. This is actually a picture of a human cell, and the area outside the cell that is shown here filled up with proteins and carbohydrates and lipids is referred to as the extracellular matrix. Right, but generally, there may not be a matrix present if you're talking about bacteria or yeast or something like that. And then, as we said, the area inside the cell is referred to as the cytoplasm or the cytoplasmic side of the plasma membrane. Okay, so remember, all organisms have a plasma membrane, but only plants and some types of bacteria have a cell wall. So all organisms have a plasma membrane, but only some organisms, namely plants, have a cell wall that's made of cellulose, and some bacteria have a cell wall that's made out of peptidoglycan. Some bacteria have 2 plasma membranes, and the space in between the plasma membranes is referred to as the periplasmic space or as the periplasm. Right? So the inside of the bacterial cell is always referred to as the cytoplasm. And if it happens to be one of the types of bacteria that has two phospholipid bilayers with a space in between, it's referred to as the periplasm. What about the nucleus? Again, remember that not all organisms have a nucleus. right? If you have a nucleus, the purpose of the nucleus is to keep the genetic material, the DNA, in a separate compartment, right? Now, in eukaryotes, they have a nucleus, but bacterial cells, even though they don't have a nucleus, they have an area within the cytoplasm where the chromosome is normally found. And that area where you find the chromosome that a bacteria has is referred to as a nucleoid. Right? So if you're a bacteria, you don't have a nucleus, but you do have a nucleoid. That's just a part of an area in the cytoplasm where the bacterial chromosome is usually found. Okay, now eukaryotes have what's known as an endomembrane system. Endo, of course, means inside. It's a Latin word for inside. And then membrane, you know what that is. So things like the endoplasmic reticulum, the rough endoplasmic reticulum and the smooth endoplasmic reticulum, that is referred to as an endomembrane system, a network of membranes that you find inside the cell as opposed to the membrane that you find surrounding the cell. Okay, so the nucleus is considered to be part of the endomembrane system of a eukaryote because the nucleus is surrounded by its own membrane, which is referred to as the nuclear membrane. And the nuclear membrane is also a phospholipid bilayer. Right, so the nuclear membrane is also a phospholipid bilayer, which is used to surround and define where the nucleus is. The liquid and the mixture of liquids and proteins and carbohydrates that you find inside the nucleus is referred to as the nucleoplasm. Remember, we referred to the space inside the plasma membrane as the cytoplasm. And then at the center of the cell, we have the nucleus, and the area inside the nucleus is referred to as the nucleoplasm. Then there's a network of little filaments that kind of holds things in their proper place inside the nucleus, and that's referred to as the nuclear matrix. And there is kind of a network of fibers that surrounds the inner part of the nuclear membrane, which is called the nuclear lamina. Those of you that have taken Biology 200 know that the nuclear lamina is very important for turning genes on and off. It's critical to the nuclear lamina is critical to the proper functioning of the nucleus. Okay, now remember that some organisms are haploid and other organisms are diploid. Okay, so haploid and diploid refers to how many copies of the complete genome does each cell have. Okay, so a genome refers to all the genes that an organism is supposed to have. So there's a human genome, there is a fruit fly genome, there's a bacterial genome, and so on. So how many genes are there in the human genome? If you don't know the answer, it's approximately 23,000. Right? And so there are about 23,000 genes in the human genome. Humans are diploid, meaning that we have 2 copies of the complete genome, so that for each of the 23,000 genes, we have two of each inside every cell. Okay, so that's what a diploid organism is. A haploid organism is something like bacteria that has only 1 copy of each gene. Right? So there is a bacterial genome, depending on which bacteria you're talking about. So Escherichia coli, for instance, there is an Escherichia coli genome, which contains about 4,000 genes, I believe. And there's only 1 copy of each of those genes. Okay, now there's another slight difference that's interesting between bacteria and eukaryotic cells, for instance, which is that eukaryotes, like humans, have what are called linear chromosomes, whereas bacteria have circular chromosomes. In fact, they have only 1 chromosome, and it is circular. Okay, so a linear chromosome means that the chromosome is rod-shaped. It's shaped like a little rod. In fact, that rod is a compressed DNA double helix. So each chromosome is made of a long thread of DNA, and when the cell is in the process of dividing, that long thread of DNA will wind together and compress itself into a kind of a little stick shape. And then the cell will divide, and the sister chromatids will go to either side of the cell. You probably remember some of that from first year. So humans and most other organisms have linear chromosomes, but bacteria have circular chromosomes. So there is no loose end. If any of you have taken Biology 200, you'll remember that the purpose of there's a structure, there's a cap on both ends of a linear chromosome called a telomere, and one of the purposes of the telomere is to prevent the chromosome from being chewed up and destroyed by enzymes called nuclease enzymes. There are, believe it or not, there are enzymes inside the cell whose job it is to destroy DNA. The purpose of that is really it's a defense mechanism so that if viruses or other cells invade your cells, these nucleases will go to work chewing up and destroying the chromosomes of whatever it is that invaded you. Okay, then how do you prevent your own chromosomes from being chewed up and destroyed by those nucleases? Well, as it happens, those nucleases are they're referred to as exonucleases because they only have the ability to start from a loose end. So they start eating up and destroying the chromosome from the loose end, right? So how does a human prevent their chromosomes from being chewed up by these exonucleases? They protect the loose end with a structure called a telomere. Okay, what about bacteria? Well, they do something that's simpler than that. They just don't have a loose end. The chromosome is a circle instead of a linear chromosome. Okay, so bacteria have circular chromosomes, and generally they have only one large, very large circular chromosome that is located in the area of the cytoplasm called the nucleolid. Many bacteria have small accessory chromosomes that are referred to as plasmids, and we'll talk about those later. But generally, bacteria, they have one large circular chromosome, and they may have one or two or a few of these little spare chromosomes that are called plasmids. All right, now, is the genome of whatever organism we're talking about, does it have a DNA or an RNA genome to encode its genes? Well, in fact, every organism on Earth has a DNA genome except one, and those are some of the viruses. So there are some viruses that have an RNA genome, but everything else on Earth uses a DNA genome. So there are some viruses like the COVID-19, the coronavirus that is keeping us away from the classroom. The coronavirus is an example of a virus that has an RNA genome, so the genes are encoded by RNA and not by DNA. Okay, next, we mentioned the fact that there are some weird-looking protists that are still around that may have more than one nucleus. Right? And so do you have a nucleus or not? Humans, obviously, human cells have a single nucleus, but there are lots of eukaryotes around, and they are all protists. They're all classified as protists that sometimes have two nuclei, depending on the species of the protist. So there are some protists like Giardia lamblia that have two nuclei, and there are some protists like Plasmodium vivax that only have one. All right, so there in our diagram, there is the nucleus located at the center of a cell. So obviously what we're looking at here is not a prokaryote. It must be a eukaryote, and it can't be one of those strange protists that has two nuclei. We only can see one nucleus there. All right, here is an actual photograph from a microscope. This is just showing you the size difference between eukaryotes and prokaryotes. These things here are eukaryotic cells. And these little dots are bacterial cells. These are prokaryotes, right? So you can see that the eukaryote is much bigger. The eukaryotic cell is much bigger than the little prokaryote cells. And of course, this structure here in the middle is the nucleus. All right, as I mentioned, do we have linear chromosomes versus circular chromosomes? This is a human karyotype, which is made up of a bunch of linear chromosomes. So you just learned that the human genome contains around 23,000 genes, and those 23,000 genes are distributed across 23 chromosomes, 23 different chromosomes, and we number them chromosome 1, chromosome 2, chromosome 3, and so on. And because we're diploid, we have 2 of each. We have 2 of chromosome 1, and we have 2 of chromosome 2, and we have 2 of chromosome 3, and so on. So this is what human chromosomes look like. This is what linear chromosomes look like. And this is what a circular bacterial chromosome looks like, right? So it's a circle, and it's quite a bit smaller. These two photographs are not to the same scale. Okay, do you have one nucleus or do you have several? So this is Giardia lamblia, which is a protist that happens to have two nuclei because it evolved during a period of time when nature had not yet decided whether it was necessary to have two nuclei or whether one was sufficient. Obviously, in the meantime, nature decided that one nucleus is fine. We don't need to have two. But these are primitive, very primitive protists that are more than a couple of billion years old, and so they happen to have two because they kind of got in before the pattern was set. Okay, so again, some differences between bacteria and eukaryotes. Bacteria have no nucleus, as I said before. The genome is confined to an area in the cell called a nucleoid. Eukaryotic DNA is associated with proteins called nucleosomes. So if you took Biology 200, I don't want to keep bringing that course up, but it is a nice course. If you took Biology 200, you learned that the DNA inside eukaryotic nuclei is not just floating around loose. The DNA double helix is actually wrapped around little spool-shaped or little spherical structures called nucleosomes, which help the cell to organize and package the DNA. So if you're doing knitting, for instance, you have a ball of wool. The wool is wound up into a ball, which makes it easy to organize, whereas if you tried to do knitting with just a pile of wool on the floor, it would probably get tangled up and it would get tied up in knots and you'd find it very difficult to work with. But as long as the wool is wound around a spool, or if you're working with thread, if it's wound around a spool, it's easier to keep it organized. Now, bacteria do not have nucleosomes because nucleosomes evolved after bacteria. But there is a primitive form of the bacteria have a primitive form of a protein that's similar to a nucleosome, which is referred to as a nucleoid associated proteins, or NAPs, right? So eukaryotic cells have nucleosomes, bacteria have NAPs, right? So the eukaryotic cells have one or more nuclei, and they have the DNA wrapped around nucleosomes, whereas bacteria have a nucleoid instead of a nucleus, and the DNA of bacteria is associated with NAPs, nucleoid-associated proteins. And as we said, eukaryotic DNA is usually organized into linear chromosomes, right? And bacterial DNA is organized as a circular chromosome, but some bacteria have a few of these little accessory chromosomes that are much smaller, but they're also circular, and they're referred to as plasmids. So, mitochondria are the energy generating slaves that you find inside a eukaryotic cell. So, they are responsible for the mitochondria are responsible for carrying out something called aerobic respiration. Aerobic respiration means that you consume oxygen and you create carbon dioxide, and through a series of reactions that is designed to give you ATP through the breakdown of glucose. So mitochondria, their aerobic respiration is where you burn glucose and use it to generate ATP. So glucose is the main energy molecule for every cell. Most of the forms of life on Earth thrive on glucose. But glucose, if you break down glucose, you release a lot of energy. In fact, so much energy that it's not really of any use to a cell, because cells need the energy to be delivered in much smaller packages. Otherwise, it's too much. For instance, the gasoline tank, the petrol tank of an automobile contains a lot of gasoline, but if you ignited it all at once, it wouldn't be creative, it wouldn't be useful, it would be destructive. So instead, we have an engine that ignites little bits of the petrol, little bits of the gasoline over time, and that can power the automobile. So aerobic respiration is where the mitochondria break down glucose and then use the energy that's liberated to make a bunch of smaller energy molecules called ATP. That is referred to as aerobic respiration. And aerobic respiration that takes place in the mitochondria, it requires oxygen and it generates carbon dioxide. It is the breakdown of glucose and then the harvesting of the energy takes place in 3 different steps. The first step is called glycolysis, where you break glucose in half. The second step is called the Krebs cycle, where there's a series of chemical reactions that generate some other energy molecules. And then the third step is called the electron transport chain, where a series of reactions leads to what's known as oxidative phosphorylation. Oxidative phosphorylation, the third step in aerobic respiration, generates by far the most ATP. Okay, now, what is the difference between aerobic respiration and fermentation? Aerobic respiration takes place in the presence of oxygen. Aerobic respiration requires oxygen. Now, if you take 1 molecule of glucose and you break it down using aerobic respiration, you will end up generating 34 molecules of ATP. That's quite a lot. So you break down 1 molecule of glucose using aerobic respiration, you get 34 molecules of ATP. Fermentation is what some organisms have to do if there is no oxygen. So aerobic respiration requires oxygen, but there are some organisms, particularly bacteria. There's some bacteria that live in an oxygen-free environment. So when they break down glucose, they eat glucose as well, but when they break it down, they can't do aerobic respiration. They're forced to do something else, which is called fermentation. Fermentation is also known as anaerobic respiration, right? So aerobic respiration means respiration or generating energy by burning oxygen, and anaerobic means in the absence of oxygen. So there's some bacteria and other organisms that live in an area where there is no oxygen, so they have no choice but to do something called anaerobic respiration, which is also known as fermentation. And what happens here is that glucose is broken down and it's converted into an alcohol, usually ethanol, or it's converted into lactic acid. So you take glucose or lactose, another sugar, and you break it down into either ethanol or methanol or lactic acid, and you generate much less ATP by doing that. So aerobic respiration, you burn 1 molecule of glucose in the presence of oxygen, you generate 34 molecules of ATP. With fermentation, you convert the glucose into an alcohol and you create a pathetic 2 molecules of ATP per molecule of glucose, which is actually a terrible waste of all the energy that's contained in glucose, but it's the best they can do. Okay, so now there are some bacteria that live in bacteria and yeast in particular that live in oxygen-free environments, and this is what they do. And that, in fact, we exploit that when we use Saccharomyces cerevisiae to make wine. We put the Saccharomyces cerevisiae into grape juice, and then we put it all into a bottle, and then we put a cork in the bottle so that there's no air. And then in the absence of air, the Saccharomyces cerevisiae is forced to convert the glucose that's in the grape juice into ethanol. And then, in fact, what happens is the Saccharomyces cerevisiae will start eating the glucose and producing ethanol. And when the ethanol content rises to 12.5%, it actually kills the Saccharomyces cerevisiae. So the ethanol, ethanol is the Saccharomyces cerevisiae waste product, which is actually toxic to the cells that made it. And so if you have Saccharomyces cerevisiae that are carrying out anaerobic respiration in a sealed bottle full of grape juice, when it produces enough ethanol that the concentration of ethanol reaches 12.5%, that is actually poisonous to the cells that are fermenting the grape juice. So that's why you never see wine that has an alcohol content of greater than 12.5%. If you don't leave, if you basically the Saccharomyces cerevisiae, you put it in there with the bottle and it will ferment the grape juice to 12.5% alcohol and then die, and that's all you have to. You don't have to worry about controlling the concentration of alcohol in wine because it'll take care of itself. It will stop fermenting when the concentration of ethanol becomes lethal. All right, so mitochondria carry out aerobic respiration. So here's some mitochondria on our diagram. Right, these all used to be a free-living bacteria called a purple cyanobacteria, which we'll talk about later in the course. This is a kind of a three-dimensional diagram. Notice that the mitochondria has two membranes, and that's because it used to be an independent living bacteria. And the inner membrane was the membrane that belonged to the bacteria, and the outer membrane is the membrane that belongs to the host cell, belongs to the cell that the mitochondria is inside. And this is an electron microscope image of a mitochondria. You can see that the inner membrane is highly folded, which is a way of fitting more of that membrane into the confined space. Okay, so just remember that aerobic respiration takes place in three steps: glycolysis, the Krebs cycle, and the electron transport chain. And remember that aerobic respiration requires oxygen, and if you don't have access to oxygen, you have no choice but to do something called anaerobic respiration or fermentation. Now, not every organism can do fermentation. Not every organism has the ability to do anaerobic respiration. But if you have the ability to live in air, it's better to live in oxygen because you can generate more oxygen from glucose that way. So if you can live in oxygen, it's better to, right? If you then get switched to an environment where there's no oxygen and you die, that means you do not have the ability to ferment glucose to create energy. So that means you can only live in the presence of oxygen. So when you have an organism that can only live in the presence of oxygen, you refer to it as an obligate aerobe. An obligate aerobe. Obligate, from the word obliged, means when you're obliged to do something, that means you have to do it. You are forced to do it. So you are obliged, if you're an obligate aerobe, you are obliged to live in an oxygen environment, otherwise you'll die. Example, humans, right? So we can't live without oxygen or we'll die. Humans are obligate aerobes, sorry. Now there are some bacteria like Clostridium botulinum that are obligate anaerobes. They deliberately avoid oxygen because it kills them. And they generate all of their energy from fermentation and anaerobic respiration. And so oxygen would actually kill Clostridium botulinum, and so we refer to them as an obligate anaerobe, obliged to live in an anaerobic environment, so an obligate anaerobe. There are some organisms that are very lucky that have the machinery to live in either an oxygen-enriched environment or an oxygen-depleted environment. And those are referred to as facultative anaerobes. Facultative means you have the facilities to do this if you want to, right? So Saccharomyces cerevisiae is the best example of a facultative anaerobe. It prefers to live in the oxygen, in the air, and it can generate a lot more energy that way to power its functions. But if you move it into an anaerobic area, like into a bottle of wine with the cork in the bottle, it can switch to anaerobic respiration because it has the facilities to do so. So it is therefore a facultative anaerobe. Okay, this is a diagram that shows the two forms of breaking down glucose to create energy. In both cases, either whether we're talking about aerobic respiration or anaerobic respiration, the first step is always the breakdown of glucose, breaking glucose in half to create two molecules of pyruvic acid. That's called glycolysis. And then if we're talking about aerobic respiration, the pyruvic acid is sent into a series of reactions that are referred to as the Krebs cycle. The Krebs cycle is also known as the citric acid cycle in some textbooks, right? And it generates a little bit more ATP, but it also generates a lot of carbon dioxide. Right? So when humans exhale carbon dioxide, that carbon dioxide all came from the Krebs cycle that's going on inside all of our cells. And then finally, the Krebs cycle creates two other types of energy molecules called NADH and FADH2, right? And those are fed into the third phase of aerobic respiration, which is the electron transport chain, also known as oxidative phosphorylation. The electron transport chain through oxidative phosphorylation consumes oxygen, right? So you have to put oxygen into it. And then at the end of it all, at the end of all these reactions, the oxygen is converted into water, right? So the oxygen that we inhale is going to the electron transport chain. The carbon dioxide that we exhale came from the Krebs cycle. So those are two related but different functions. Now the lion's share, by far the majority of the ATP that's created from aerobic respiration is created by the electron transport chain. So you have two molecules of ATP that are created by glycolysis, and you have one that's created by the Krebs cycle, and then you have about 32 that are created by the electron transport chain. So you can see that it's definitely to your advantage to do aerobic respiration if you can. If you have the enzymes to do it, it's better to live in the air and generate lots of energy from the breakdown of each molecule of glucose. On the other hand, if you have no choice, you do glycolysis, and then you attach things to you create alcohols. You take the pyruvic acid and you convert it into ethanol or lactic acid or methanol in some cases, and then that's it. You generate just 2 molecules of ATP. That's not very much, but what choice do you have? If you don't have oxygen, you can't do aerobic respiration. Okay, so glycolysis, step 1. The Krebs cycle, step 2. The electron transport chain, step 3. Right? Total 34 molecules of ATP to use for the cell to use for energy. Anaerobic respiration generates basically just 1. Okay, now these terms that I mentioned a minute ago, they still apply, right? So an obligate aerobe is an organism that can only live in the presence of oxygen. An obligate anaerobe is an organism that can only live in the absence of oxygen. And a facultative anaerobe is an organism that can live in either one because it has the facilities, it has the machinery or the facilities to do either one. Example of an obligate aerobe would be humans. There's several bacteria that are obligate aerobes as well, and we'll get into that. There are many bacteria that are obligate anaerobes, like Clostridium botulinum or Clostridium perfringens, for instance. And there are many organisms that are facultative anaerobes. The best example that we have is Saccharomyces cerevisiae. Okay, I mentioned the fact that mitochondria used to be independent bacteria that were swallowed at some point in history by eukaryotic cells. Probably a couple billion years ago that happened. And this is known as the endosymbiotic hypothesis, or the endosymbiotic theory, that eukaryotes, a eukaryotic cell at some point swallowed bacteria that happened to be producing a lot of excess energy, producing a lot of excess ATP, and instead of digesting them, the cells accidentally just kept them, and then this turned out to be an advantage because they were generating so much spare electricity that this was a great benefit to the host cell. Now, what is the evidence for the endosymbiotic hypothesis? Well, number 1, we just mentioned that eukaryotes have linear chromosomes and bacteria have circular chromosomes. All right, if you look at a mitochondria, if you look deep inside a mitochondria, you will see that it has a single circular chromosome of its own, right? So the genes that encode the proteins that you find inside a mitochondrion are encoded by a circular chromosome that the mitochondria has. Okay. Second piece of evidence, bacteria have smaller ribosomes than eukaryotes. Eukaryotes and prokaryotes have different types of ribosomes. Eukaryotes have what's known as an 80S ribosome. Regular bacteria have what's known as a 70S ribosome, and a very primitive type of bacteria called an archaebacteria has a 55S ribosome. It turns out that if you look at the ribosomes that you find inside mitochondria, they are 55S in size, right? So these mitochondria, they have a circular chromosome, they have a 55S mitochondria, and they have their own membrane. They have their own internal membrane. And those things combined indicate or suggest to us that they used to be bacteria because they look just like archaebacteria. In fact, they look like a specific type of archaebacteria called purple cyanobacteria that we'll talk about later in the course. The endomembrane system. Let's talk about that. That is the rough endoplasmic reticulum, the smooth endoplasmic reticulum, and the Golgi apparatus. Okay, so the rough ER, the RER, and the smooth ER. The rough ER, as you remember from first year, is there. It's involved in the synthesis of proteins that are meant to be exported out of the cell. The smooth endoplasmic reticulum is involved in the creation of lipids. The Golgi apparatus is responsible for glycosylating proteins that are going to be sent out. So any protein that is destined to be exported from the cell through exocytosis, it will be created or translated inside the lumen of the rough endoplasmic reticulum. Remember the term lumen, L-U-M-E-N. L-U-M-E-N, lumen means the hollow compartment inside something. That's the general term. Right, so the proteins that are meant to be exported out of the cell will first be translated in the lumen of the rough endoplasmic reticulum, and then they will be sent to the Golgi apparatus where they receive carbohydrates. And so if you put carbohydrates onto a protein, that's referred to as glycosylation, and you have created glycoproteins. And they are then sent out, and the glycoproteins act as sort of an address. Like if you send a glycoprotein out into the blood, it will bump into other cells, and those other cells will decide whether or not to let it inside. Whether or not they decide to let it inside depends on what carbohydrates are attached to it. Right, so glycosylation is kind of like a post office where you put addresses on parcels that you're going to send out into the environment to go someplace else. So that's what the Golgi apparatus does, or the Golgi complex. All right, so here we have the rough endoplasmic reticulum, and over here we have the Golgi complex in this diagram of a cell. Here is a picture. This is an actual transmission electron microscope image. All of this stuff here is rough endoplasmic reticulum. This is the nucleus of the cell here, right? And then all of this stuff is rough endoplasmic reticulum. It's called rough endoplasmic reticulum because it has ribosomes stuck to the outside. These little black dots that you find all over the outside of the rough ER, those are ribosomes, and they give the endoplasmic reticulum the appearance of being rough. Like if you were able to touch it, it would feel rough to the touch because there are all these ribosomes stuck to the outside. All right, so here's a diagram, a cartoon diagram of a cell. There's the rough endoplasmic reticulum where these proteins that are meant to be exported out of the cell are being created. Then they are moved through things called transport vesicles to the Golgi complex, and then in the Golgi complex they are glycosylated and then sent out of the cell through exocytosis. Here's a transmission electron microscope of the Golgi complex. Ribosomes. Ribosomes are responsible for the synthesis of proteins. The synthesis of proteins is where you read a messenger RNA and then you convert it into a protein. That process is called translation. Right? So if you have genes that are made of DNA and they're located in the nucleus, what you do if you want to convert those genes into protein is you make a temporary copy of the DNA using a temporary material called RNA. So instead of deoxyribonucleic acid, you make a temporary copy of ribonucleic acid, and then you send that out to the cytoplasm where it gets loaded onto a ribosome and then translated into protein. Okay, so here's a ribosome. It looks sort of like that. It's composed of two different subunits. There's a large one, the large subunit, and a small one, the small subunit. The large and the small subunits of the ribosomes don't actually come together until they are attached to a ribosomal messenger RNA. Okay, so the two subunits in eukaryotes, the ribosomes are what are known as ADS ribosomes, and that the ADS ribosome is composed of a 40S and a 60S. Okay, now the units, the unit stands for Svedberg unit, which is a kind of a it's a measurement unit unlike any other measurement unit, which is based on a technique called centrifugation. So if you took biology 200, you know what that means. It means that you spin the ribosome in a centrifuge and you see how fast it travels downwards through the tube, and the speed at which it migrates downwards through the tube as a result of centrifugal force. That's measured in something called Svedberg units after the scientist who invented this method of measuring the sizes of ribosomes. So you notice that ADS, that's a fairly large ribosome, is composed of a 40S subunit and a 60S subunit, and 40 and 60 do not add up to 80, they add up to 100. So why are we calling it an ADS ribosome? It's because the units are not additive. So if you put together the 40S, if you spin the 40S subunit and the 60S subunit through a centrifuge, they will spin through the centrifuge at a certain rate. If you put them together, they will spin through the centrifuge at a greater rate, but not in an additive fashion, right? Okay, so eukaryotes have 80S ribosomes, and prokaryotes have 70S ribosomes, which is composed of a 30S and a 50S subunit, Svedberg units again. And there are some very primitive types of bacteria called archaebacteria that are billions of years old, and they have what's known as a 55S ribosome, which is composed of a 28S and a 39S ribosome, right? And so eukaryotes have those 80S ribosomes. Regular bacteria, modern bacteria, have 70S ribosomes. And primitive bacteria have 55S ribosomes, which is also what mitochondria have. Okay, now why do we care so much about ribosomes? One of the reasons why we care about them for this course is because if you get infected with bacteria, we have to have some way to kill the bacteria that will not kill you. Right? That's the whole trick of curing infectious microbial diseases. You have to find some chemical or some drug that will kill the bacteria without killing you. In order to do that, you have to look for differences between the bacteria and the human host. And one of the differences is, of course, the ribosomes. So there are drugs that specifically kill bacteria, and we call those antibiotics. Now, antibiotics do nothing against viruses. They do nothing against protists or protozoa. They do nothing against helminths. They do nothing against viruses, right? They only kill bacteria, right? So sometimes people mistakenly go to the doctor and ask the doctor for antibiotics when they've got a cold. Well, that's foolish because a cold is caused by a virus and an antibiotic will only kill bacteria. So what do you need antibiotics for? And so that's something called antibiotic abuse. The doctor is supposed to say, no, you can't have antibiotics for a cold or a flu because antibiotics kill bacteria and colds and flus are caused by viruses, so you can't have antibiotics. But sometimes the doctor, unfortunately, sometimes the doctor will just get tired of hearing you're asking for antibiotics, and they will write you a prescription for antibiotics just to get you out of their office, and they're not supposed to do that. And that is something that's known as antibiotic abuse. The liberal use of antibiotics in situations where you don't need them and where they don't do any good, and this is what's causing what are known as superbugs to arise. Okay, so antibiotics kill bacteria. Antibiotics kill bacteria. The antibiotics have to kill the bacteria without killing the human host. And so there are many antibiotics that target the differences between human and bacterial ribosomes, right? So many antibiotics target the bacterial 70S ribosome while leaving the human 80S ribosome alone, right? So the 70S ribosome that a bacteria has is a common target of antibiotics. So remember that, because that's a question that I like to ask on exams. All right. Now, you know that a vessel like a ship is something that carries cargo from one place to another. If you have microscopic vessels inside cells, we refer to them as vesicles. Instead of vessels, we call them vesicles. So vesicles are little tiny containers that carry things from place to place inside a cell. There are two that we're going to learn about. One type of vesicle is called a lysosome, and the word lyso or lysis is a Greek word meaning to tear apart or to break apart. Okay, so lysosomes are little tiny vesicles that contain digestive enzymes whose job it is to break down certain proteins or lipids or carbohydrates that are brought into the cell through endocytosis. Okay, so lysosomes are used to facilitate cells eating things. Right? Peroxisomes are little vesicles that contain enzymes that are involved in detoxifying toxic chemicals, in particular hydrogen peroxide. So we just finished talking about the mitochondria, and we talked about the fact that the mitochondria need lots of oxygen. Now, whenever you need oxygen, whenever you take oxygen and you mix oxygen with water, you generate, unintentionally, you generate hydrogen peroxide. And hydrogen peroxide is a very reactive chemical. It breaks proteins, it kills the activity of proteins, something that's referred to as protein denaturation. So hydrogen peroxide is a toxin. But there is an enzyme called peroxidase that is able to convert hydrogen peroxide into gaseous oxygen and water. Right? So if you mix gaseous oxygen and water together, you sometimes create hydrogen peroxide. The formula for hydrogen peroxide is H2O2. It's a dangerous free radical, but there is an enzyme called peroxidase that causes the H2O2 to go back into the form of H2O and O2. So those enzymes are contained inside little vesicles that are called peroxisomes. There are some other enzymes in there as well that detoxify other chemicals, but one of the main enzymes that you find inside a peroxisome are the hydrogen peroxidase enzymes. Okay, and when you bring food into the cell, when a cell swallows a piece of food, that's referred to as phagocytosis. We should remember that from first year. And phagocytosis, you bring food in and put it in a vesicle. That vesicle will then fuse with a lysosome so that the digestive enzymes that are contained inside the lysosome will be mixed with the food, and then it can break the food down. Those digestive enzymes will break down the food so that you can break them down, break proteins down into amino acids, and then the cell will use the amino acids to make new proteins, and so on. Okay, some microbes have a large empty space at the center, which is referred to as a central vacuole. Right? Vacuous, the term vacuous means empty. Right? So a vacuole is a large empty container in the center of many types of microbes. You find central vacuoles in the center of plant cells, and you find central vacuoles in the center of a number of different protists, and we'll talk about those later. But a central vacuole is another type of a vesicle, basically. So here we have a diagram showing food getting taken into a cell through phagocytosis. That food then fuses to a lysosome. It docks with a lysosome. The lysosome releases the digestive enzymes inside the food vacuole, and it breaks down the digestive enzymes then break down any proteins or lipids or carbohydrates into amino acids or fatty acids or into monosaccharides. And those components can then be used again. It's like if you had a house and you tore down the house, but you kept all of the big beams and some of the other things and used those things, recycled those components to build another house. Okay, so lysosomes can be distinguished under the microscope because they're kind of dark colored. Peroxisomes can be distinguished because, well, we'll see them in a second. Okay, this is a central vacuole which is found inside of a very neat, interesting kind of protist called a paramecium. And this central vacuole is filled with water, and the paramecium live in water. And they have this central vacuole in the center of their body that is filled with water, and they can make that central vacuole bigger or smaller. And they do this in order to go up or down in the water. If they want to go deeper into the water, they suck some more water into the central vacuole. The central vacuole gets bigger, and the paramecium sinks down lower into the water. If they squeeze some of the water out of the central vacuole, they float upwards towards the top, which, by the way, is exactly the way a submarine goes up and down in the water. You have ballast tanks, and you squeeze, you let water into the ballast tanks, and the submarine goes down. You push the water back out with compressed air. The submarine goes back up. So that's basically what a paramecium is using the central vacuole for. All right, let's talk about the fact that inside of a cell, instead of just having liquid inside a cell, you also have a number of little different things, filamentous things. Filament means a little straw or a rigid little thread of something. That's a filament, right? So there are inside a cell is not just an amorphous blob. It's not something that has no shape. Generally, all cells are held into a certain shape by a skeleton, a little tiny microscopic skeleton, which is referred to as a cytoskeleton, right? And then the bones of that skeleton are little protein filaments, little filaments made of protein that we refer to as cytoskeletal filaments. And there are 3 different types. They are classified according to their size. The smallest ones, which are only about 7 nanometers in diameter, are called microfilaments, and they are made of a protein called actin. Then we have very large filaments that are hollow on the inside like a tube, and we call those microtubules. Microtubules contains the word tube, so that should help you to remember that they are hollow on the inside. And they are made up of a protein called tubulin. Microtubules are made of tubulin. Microtubules are the largest of the cytoskeletal filaments. They are approximately 25 nanometers in diameter, so they're more than twice the size of the microfilaments. And then in between, we have filaments that are of intermediate size, and we literally call those intermediate filaments. And they are 10 to 12 nanometers in diameter, right? So the microfilaments the actin microfilaments are about 7 nanometers in diameter, and they are not hollow. The microtubules are 25 nanometers in diameter, and they are hollow on the inside, therefore they form tubes. And the intermediate filaments are 10 or 12 nanometers in diameter, and they are not hollow inside either. Why did I not list a protein that they're made out of? Because there are several different proteins that form intermediate filaments. One of the proteins is keratin, which is also what hair and fingernails are made of. Another substance that some of them are made out of is called lamin B protein, and lamin A protein, and lamin C protein, but don't worry about that for this course. So here we see a diagram. Here we have the microtubules, which are the largest, and then the microtubules in this image here are stained in green, so you can see where they all are. These are the actin microfilaments. This is another cell where we have stained the actin microfilaments, so you can see where they are. And here are some intermediate filaments that are stained in green, and you can see where they are throughout the cell. Here they are together. The green stain is the microtubules, so there's quite a few microtubules inside a typical. This is a eukaryotic cell. This is a human. I believe it's a human fibroblast. So this is a typical human cell where we have visualized all of the cytoskeletal filaments. So the green ones are the microtubules, and the red ones are the actin microfilaments. So you can see how extensive in a cell, how extensively the cell is held into a certain shape by cytoskeletal filaments. A cell is not just a bag of water or a bag of liquid. It is actually held into a very specific rigid shape by a lot of these cytoskeletal filaments. Propulsion. How do cells move around? Not every cell moves around, obviously, but some of them do. Okay, now there are some cells that are independent cells, particularly protists, protozoa, that are covered on the outside with little finger-like projections called cilia. Cilium is singular, cilia is plural. Right? And the cilia are below the plasma membrane. So you have cilia with plasma membrane on the outside. Okay, now there are some cells in the human body that are ciliated as well. They have cilia on the outside, right? And if you've taken biology 120 or 130 or biology 200, you should be able to tell me which cells in the human body are ciliated. Okay, I'll give you a minute to think about that, right? And I'll tell you the answer, right? The trachea is the cells that line the trachea are ciliated. And the cells that line the fallopian tubes, the oviducts in women, are also ciliated. So for example, the cilia in the oviducts, what they are doing is they are moving the egg down the oviduct from the ovary to the uterus. Right? So these motorized little fingers are deliberately moving an egg from the ovary to the uterus. Okay, but there are some cells that are independent cells, like I just showed you a paramecium. Paramecia swim because their body is completely covered with cilia. In fact, the paramecium, another name for paramecium, they are also known as the ciliates. They're members of a family called the ciliates because they're completely covered with cilia. Okay, so cilia is generally a propulsion system that's used by protists. It's used by eukaryotic cells. Bacteria generally don't propel themselves with cilia. They propel themselves with a little whip structure that's called a flagellum, and we'll talk about that next. But before I leave the cilia, the interesting thing about cilia is that they can be used by a protist to swim, but they are covered on the outside by the plasma membrane. Now, the immune system, the human immune system, if you get infected with a protozoa or a bacteria, the human immune system will get to work and try and kill those invaders. The human immune system is able to recognize your proteins from invading proteins. It's able to recognize the proteins that are on your cells and distinguish the proteins that are on your cells from the proteins that are on an invading protist or an invading bacteria. Now, the cilia, so the human immune system is very good at being able to recognize foreign proteins that are present on an invading protist, for instance. But if that protist, the cilia are made of basically microtubules that move around, but they're covered up on the outside by a phospholipid bilayer. The human immune system is not very good at spotting or recognizing foreign phospholipids because they look just like our phospholipids. So by covering your cilia in a phospholipid bilayer, you're actually able to hide from the human immune system. So many of these protozoa that get inside our body are they're swimming around with the help of cilia, but they're hiding from the immune system because they've covered their cilia up with the phospholipid bilayer on the outside. Now let's discuss flagellum. The word flagellum is a Latin word meaning whip, right? So you might have heard the term to flagellate. The word flagellation means to whip with a whip, like you're punishing somebody by whipping them. So that's called flagellation. You've sometimes heard, somebody is saying bad things about themselves. Somebody will say the metaphor. They'll say you're flagellating yourself. You're flagellating yourself. That means you're whipping yourself on the back with your own hand. Okay, so a flagellum is a Latin word for a whip, and there are some microbes that swim around with the help of a flagellum. If you have flagellum is singular, flagella is plural, right? So there are many bacteria that swim around with the help of a flagellum or flagella, several of them, and flagella are made of protein, right? So if flagella are made of protein, and if our immune system is very good at recognizing foreign proteins, it stands to reason that our immune system can very easily target flagella, right? So these organisms that cover their cilia with the plasma membrane can hide, But these bacteria that are swimming around with the help of a flagellum are easier for our immune system to spot, easier for our immune system to recognize and destroy. Okay, now, how many flagella does a particular bacteria have, and where are they located? That's one of the ways that we distinguish one type of bacteria from another. If somebody is sick because they've been infected by a bacteria, you take a blood sample or something and you look at the sample under the microscope and you see that the bacteria has flagella, and it has several flagella that are located at one end of the cell. That's referred to as lophotricious when that happens, and then you can guess which bacteria that is based on how many flagella it has and where they are located. So, looking at the flagella that are located on a bacteria, does it have flagella, yes or no? If it does have flagella, does it have just one, or does it have more than one? If it has more than one, where are they located? Are they all located on the same side of the cell, or are they located all over the cell? That is one of the ways that we analyze and discover the identity of bacteria based on their morphology. All right, so here's a microscope image which has been colorized. This is something called a false color electron microscope image. You learned about that in the last lecture. So this is a protozoa that is covered with cilia that it's using to swim, and those cilia are located underneath a plasma membrane. Here's another one. This is a paramecium. That other image that I showed you of a paramecium was a paramecium under a light microscope. This is a paramecium that was taken, they took the photograph with a scanning electron microscope. Okay, so here on the left we have a protist that's covered in cilia, and on the right we have a bacteria that has 4 flagella. So the 4 flagella, these are larger appendages that it uses to swim, and it kind of flips them around like somebody using a whip. Okay, and as I said before, we often identify a bacteria based on the arrangement of the flagella. So we're going to talk about that later in one of the later lectures on bacterial morphology. But generally, if you have, for example, if you have one bacteria and it's located at one end, you refer to that bacteria as a monotricious bacteria. If you have several flagella that are located at one end only, you refer to that as lophotricious. If it has one flagella located at each side of a bacillus, that's called amphitricious. Right? If it has several flagella located at the two ends of a bacilla, it's called amphilophotricious. And if it's completely covered in flagella, it's referred to as paratricious. So we will have to learn the names of those arrangements of flagella, so you can start now if you want to, or you can wait a couple of lectures when I introduce it. But that is the whether or not a bacteria has flagella and how they are organized on the body of the bacterial cell is another one of the ways that we identify certain bacteria. All right, so that's all the cell structures. You might want to see if you can identify them by pointing them out on this slide. You can pause the video if you want. So this on the right is a plant cell because it has a cell wall, and the cell wall is kind of holding it into this polyhedral shape. In the middle you can see a big pocket that's filled with liquid. That's the central vacuole. So this is a plant cell, and also the fact that this is a plant cell is given away by these chloroplasts, right? So we have some chloroplasts here. All right, now the difference between a eukaryotic cell, which is quite large, and a prokaryotic cell, which is quite small. Look at the scale. On the left here we have a prokaryotic cell, a bacteria, that is generally between 0.1 microns and 1 micron. This scale is a little bit wrong, so that should say 1 micron, not 10. Whereas a eukaryotic cell is larger. It has an endomembrane system including membrane-bound organelles like the mitochondria, and it has a nucleus. It's quite large, and it is typically 10 to 100 microns in diameter. 100 microns in diameter is very large for a cell, so you only find protozoa that are 100 microns in size. You never find human cells that are 100 microns in size. Generally, human cells are 10 or thereabouts, 10 microns in diameter or thereabouts. Okay, again, notice the difference in size. These large things are eukaryotic cells with a nucleus, and these little spots are actually bacteria. So the bacteria are quite a bit smaller than the eukaryote. Okay, so we have looked at all the different organelles. We've learned in detail about the difference between prokaryotes and eukaryotes. We've learned the difference between single-celled and multicellular organisms. We've learned about the plasma membrane. We've learned about the nucleus and the shapes of the chromosomes. We know what the difference between haploid and diploid organisms is. We know what the mitochondria are. We know where they came from. We know what aerobic versus anaerobic respiration are and fermentation. We know about the difference between an obligate aerobe, a facultative anaerobe, and an obligate anaerobe, right? That depends on whether you can live with you can only live in an oxygen environment, versus only living in an oxygen-free environment versus being able to do both, which is what a facultative anaerobe can do. All right, so then we learned about ribosomes, we learned about different types of vesicles, and we learned about different types of propulsion machines, including cilia and flagella. Okay, let's again review some of the different types of microorganisms that we're studying in this course, and let's look at the difference between a bacteria, a protist, a fungus, a helminth, and so on. Okay, so as we said earlier, biology is the study of living things that are big enough to see without a microscope, and microbiology is the study of living things that are small enough that you need a microscope to actually see them. Okay, so prokaryotes are bacteria. They do not keep their genetic material in the nucleus. They are always haploid. They usually reproduce asexually, which means that you do not find male and female bacteria. There is a type of bacterial sexual reproduction that's referred to as conjugation, and we'll talk about that much later, but it's not really sexual reproduction the way higher animals do it. They are quite a bit smaller than eukaryotic cells, and they always exist as single-celled organisms. You never find bacteria forming a multicellular organism. Eukaryotes keep their genes inside a nucleus, which has its own nuclear membrane. They are usually diploid. There are one or two examples of eukaryotic cells that are haploid, but that's quite rare. But there are also eukaryotic cells that are triploid or tetraploid. Usually those are plants, and we're not going to worry about those in this course. Prokaryotes may reproduce sexually, where there's a male and a female type of cell or a male and a female type of organism. That's obviously what humans do. They're generally quite a bit larger than prokaryotes, and many of them are multicellular instead of just single cellular. Okay, so bacteria, single-celled haploid prokaryotes. Archaebacteria are very primitive forms of bacteria that evolved when the Earth was quite different. Back when the Earth was a lot hotter and it had a lot more hydrogen in the atmosphere and a lot less oxygen. Some of those archaebacteria that breathe and they decompose sulfur, for instance. They decompose hydrogen. They produce methane gas. Those types of bacteria were around 2 billion years ago when the Earth looked a lot different than it does now. But some of them are still around today in areas that look like the primitive Earth. So you still find archaebacteria around volcanic vents on the ocean floor because there's a high temperature and a lot of sulfur there, and so they're still managing to survive there. They're not completely extinct, but they are rare and they're quite a bit different than regular bacteria. Okay, so we have bacteria and we have archaebacteria. Archae means old, primitive. Okay, we have fungi. Fungi are classified as eukaryotes. Some of them are single-celled and some of them are multicellular depending on conditions. Generally, the main condition which determines whether a fungi is a multicellular organism or a single-celled organism is the temperature. Right? So usually at higher temperatures, fungi exist as individual cells. At lower temperatures, they exist as colonies called mycelia, fungal colonies. Yeast are single-celled fungi, basically fungi that are always single-celled. Protozoa are large, elaborate, single-celled eukaryotes. Protozoa is not quite the correct word to use. We generally like to use the word protist. What is the difference? Whether or not they can move on their own. right? So if they can move on their own, if they can swim with the help of cilia, or if they can crawl around the way amoeba do, we refer to them as protozoa, because the word zoa means the ability to move, the ability to propel yourself. But there are some large single-celled eukaryotes that are classified as protists, and they can't move. They don't have flagella, they don't have cilia, they can't crawl, but nevertheless they are still single-celled eukaryotes, large single-celled eukaryotes. So collectively, we refer to the protists, that we refer to them as protists. We refer to these organisms as protists, and specifically, if they are able to move on their own by swimming or crawling, we call them protozoa. But there are a lot of protists that can't move on their own. There are a lot of protists in the ocean, for instance, that they do not swim, they simply get carried around by the tide. So there are lots of those. They are classified as protists, but they're not protozoa. They're not classified as protozoa. Okay, helminths are large multicellular parasitic worms, and viruses are not actually living things. They are a few rogue genes that are located inside a capsule called a capsid, that they invade a cell and they kind of take over that cell and force that cell to use its metabolic machinery to make more viruses. Okay, bacteria and archaebacteria. Example, this is E. coli, Escherichia coli. How would you describe the morphology of these cells? We discussed it in the previous lecture. Right, so these are rods or bacilli. Okay, so this is Escherichia coli. These are single-celled bacilli. They happen to be paratricious, meaning that they are completely covered with flagella, but those flagella are not visible unless you stain them with a special stain, which we'll talk about later. Okay, fungi and yeast. Example, here we have Saccharomyces cerevisiae. So Saccharomyces cerevisiae is a yeast. We use it to brew beer and wine, but it's not just used for that. You find it all over the place naturally. You know, every square centimeter of human skin is covered with thousands of Saccharomyces cerevisiae cells. No matter how hard you wash your hands, you still have some Saccharomyces cerevisiae there. Okay, this is another yeast called Candida albicans, and it is a dimorphic. It's a dimorphic yeast that at very cold temperatures it can form a hypha, which is a little strand, but generally it's a yeast that just exists as a single cell. And as you remember from the previous lecture, Candida albicans is the cause of a disease called vulvovaginal candidiasis, which is commonly known as a yeast infection in women. Okay, here's a fungi which is more complicated. This is called Amanita ocreata, which is a poisonous toadstool. But mushrooms and toadstools are examples of fungi, just the same way that moldy bread is an example of fungi as well. Okay, so here are some mushrooms growing. This is actually, you don't actually see mushrooms except when two fungal colonies mate. Right, so these mushrooms are growing in something called a fairy circle, because, you know, in the Middle Ages, people used to think that a bunch of invisible fairies had been dancing in a circle there, and they left these mushrooms behind when they left. So that's actually the result. A fairy circle like this is a result of two very large fungal colonies underground coming together, and the area where they overlap is where these mushrooms grow. These mushrooms that are growing above ground are known as a fruiting body, which is a fruiting body is where the spores are produced. So we'll learn how to spell fruiting body later on. Okay, here's a protozoa, Giardia lamblia. This is a scanning electron microscope image of it. Here's another one, Trypanosoma brucei, which causes a disease called sleeping sickness. These are beautiful ocean protists, which do not swim around, so we can't call them protozoa. One of them is called a radiolarian. The other one is called a diatom. We'll talk about those when we get to it. Helminths are multicellular parasitic worms. This is a multicellular parasitic worm called Tanea saginata, also known as the beef tapeworm. This is an electron microscope image of Tenea saginata's head, which is referred to as a scolex. We'll talk about that later. And it is very small, but when a Tenea saginata grabs hold of the inside of your intestine and starts absorbing your food, and it gets bigger and bigger until it becomes enormous. And so that little thing in the bottom is a 1 foot ruler, and you can see that this is a Tenea saginata that was pulled out of somebody's intestine surgically, and you can see that it's about 10 meters in length. So they start out small, but they can grow to be very large. Okay, this is a virus. This is a rhinovirus, and this is actually a computer-generated image of a rhinovirus. So it's not actually a living thing. It's some genes that are contained in a capsule that goes around from cell to cell, hijacking or usurping the metabolic machinery of that cell and using it for its own foul purposes to make more viruses. Okay, so this is human rhinovirus, belongs to a certain family. We'll talk about the different families of viruses. Human rhinoviruses cause colds. They have an RNA genome. So I mentioned the fact that viruses are the only organisms on Earth that have RNA genomes instead of DNA genomes, and only a few of them have an RNA genome. There are also a lot of viruses that have a DNA genome. And when I said organisms, viruses are not actually organisms. I'm just using that for purposes of, what else do you call them? They're not actually a living thing, but we'll call them organisms. And the fact that people who study viruses call themselves virologists, but they're also called microbiologists, right? So what is the biology of a virus? Well, actually, it doesn't have any biology until it invades a cell, but nevertheless, we'll call it an organism just for purposes of expedience. Okay, so how big are these things? We mentioned earlier that if you take a meter stick and you divide it by 100, you have a centimeter. That's also known as 10 to the minus 2 meters. If you divide it by 1,000, you have a millimeter. And if you take 1 meter and you divide it by a million, you have 1 micron, or 1 micrometer, or micron. If you divide a meter by a billion, rather, if you divide it by a billion, you have 1 times 10 to the minus ninth meters, and that is called a nanometer. So generally, when we're measuring the sizes of cells, we talk about the sizes of cells being in microns. Typical bacteria is 1 micron. Typical human cell is 10 microns. Typical protist is 100 to 500 microns. Typical yeast is 5 microns. Typical yeast cell is 5 microns. And then we discussed the size of the organelles, generally in nanometers. So you already learned that the plasma membrane is about 5 nanometers in thickness. A ribosome is about 5 nanometers tall. The nucleus is quite large. The nucleus of a cell is about 2 microns or 3 microns. And the mitochondria are quite large because they used to be bacteria, and they are typically 1 micron. Okay, so how big are these organisms in general? So viruses are very small. So viruses can be anywhere from 10 nanometers to 100 nanometers, right? And so they are the size of an organelle. They're not much bigger than a ribosome inside a cell. Bacteria, typically 1 micron in size. Archaebacteria are also 1 micron in size. As I mentioned, the mitochondria are 1 micron in size, and that's not a coincidence. It's because they used to be bacteria. Yeast cells are typically 5 microns in size. Fungi are the size of a yeast cell, 5 to 10 microns. Protozoa tend to be larger, the protists, and then the helminths are anywhere from 10 microns to 10 meters, depending on what stage of their life cycle they're in. Okay, so I'm just going to quickly go through this. In Appendix B of your textbook, there's some useful resources for you to look things up if you're ever interested in looking things up, because in Appendix D, they list bacteria and viruses by families, and they also list the diseases that they cause. So this course is very biased in favor of teaching clinical microbiology. Clinical microbiology is the study of bacteria and viruses and things that make us sick. General microbiology is the study of microbes in general, only a small fraction of them make us sick. But this course concentrates more on clinical microbiology than the other types of microbiology. So if you're ever interested in looking up some diseases that are caused by microbes, just look in Appendix D of your textbook. Okay, so if you look in Appendix D, you'll find all of these organisms listed. So two examples of viruses that you should probably memorize as soon as possible. One is called varicella zoster virus, or VSV, which causes the disease called chickenpox if you're young. Pox, of course, are little red spots. So varicella zoster virus causes chickenpox when you're young, and it causes a disease called shingles that you get when you're middle age. So if you had chickenpox when you were a child, there's a very strong possibility that you will have shingles when you're an adult. And the reason for that is because varicella zoster is what's known as a latent virus. The word latent means that when something becomes latent, it means it goes away and hides and doesn't make any noise and it doesn't make a fuss. It just hides for decades and then it reappears and it causes a fuss. And so varicella zoster virus is a latent virus that causes chickenpox when you're young, so it's causing a fuss, it's causing you to feel sick and be covered in red spots. And then it goes silent for a few decades, and then it returns when you're a middle-aged person, and it causes blistering of the skin, which is something called shingles. All right, another virus that you should memorize immediately is hepatitis A virus, which causes hepatitis. The word hepato refers to the liver, and itis means inflammation. So hepatitis simply means an inflamed liver. So the hepatitis A virus is a virus that infects liver cells and causes hepatitis. Okay, some of the bacteria that we're going to learn that you should memorize as soon as possible. Lactobacillus acidophilus. I can already tell you, just by looking at the name, you should be able to tell me what shape the cells are. They are bacilli. And you should be able to tell me that they are acidophiles. They produce acid and they like to live in an environment that has a low pH. Now, you would find lactobacillus inside the human vagina, inside the human intestine, and you also find it inside yogurt because lactobacillus acidophilus is used to ferment the lactose that's in milk and turn it into lactic acid. That's why yogurt has kind of a sour taste, because anything that's acidic has kind of a sour taste. Okay, but Clostridium botulinum is a soil organism. It is an obligate anaerobe, and it causes botulism, or food poisoning, which we learned about in the last lecture. A couple of examples of yeast. Saccharomyces cerevisiae is present on the skin. It's part of the normal flora of the skin. You also find it on the skins of grapes, right? So you naturally find Saccharomyces cerevisiae on the outside of grapes, which explains how the fact that the early French and the early Romans knew how to make wine by simply they would stomp on the grapes with their feet, or they would use a grape press, and then the grape juice would temporarily come into contact with the grape skins, right? And so that's how the Saccharomyces cerevisiae would get into the grape juice, because it was already on the grape skins. You also have Saccharomyces cerevisiae on human skin. You also have it a lot of other places. Okay, and then so Saccharomyces cerevisiae is a naturally occurring yeast that we have learned how to make use of. We learned over many years that you can make wine if you take grape juice and you actually inoculate it with Saccharomyces cerevisiae. To inoculate means to deliberately infect it, right? So you take grape juice, and these days at the wine producing plant, they're not stomping on the grapes with their feet. They do squeeze them in a big press, but they don't actually rely on yeast. They don't actually rely on Saccharomyces cerevisiae being on the skin of the grapes anymore. There is some there, but they actually take a big handful. They take several handfuls, or they put all the grape juice in a very large vat, and then they throw in a kilogram or so of Saccharomyces cerevisiae deliberately to start fermenting the grape juice. And so in the old days, whichever strain of yeast got into the wine, it was a little bit haphazard. It was a little bit of an accident. And it turned out that the places in the world that made the best wine just happened to have a lot of Saccharomyces cerevisiae around the area, so that was on the grape skins, and so that happened to be the yeast that got into the grape juice. As soon as we learned that, we started deliberately putting the Saccharomyces cerevisiae in there. Whereas there were lots of places in the world that would make lousy wine, and the reason the wine tasted so bad was because it wasn't Saccharomyces cerevisiae that was on the outside of the grape. It might have been Candida albicans. So yes, indeed, you can actually make wine if you take grape juice and you inoculate it with Candida albicans. It would taste awful because Candida albicans doesn't produce lots of nice ethanol the way Saccharomyces cerevisiae does. Candida albicans produces a lot of other things, including acids and so on. So the wine would taste terrible, but you could theoretically make wine by inoculating grape juice with Candida albicans. However, for this course, we know Candida albicans as a fungus that's on the skin. Normally, it's part of the normal skin flora, and if it manages to travel from the outer surface of the skin inside the vagina, what will normally happen to it? Under normal circumstances, the Candida albicans will be killed or excluded by the acid in the vagina that is being created by the Lactobacillus acidophilus. But if the woman has just taken antibiotics to kill some other bacterial infection, those antibiotics will also kill the Lactobacillus acidophilus that was excluding the Candida albicans from the vagina. So that way, this is what's known as it's known as an opportunistic infection. An opportunist is somebody who takes advantage of somebody else when they're weakened or when they're not suspecting to be taken advantage of. Right? So an opportunistic infection is where you're infected by a bacteria or a yeast or some other microorganism that would not normally make you sick if you were healthy, but because you've been weakened somehow, or your body's natural balance has been disturbed, then that gives these bacteria or fungi or yeast the opportunity to get in there and start causing a fuss. Okay, so Saccharomyces cerevisiae and Candida albicans are two yeasts that you should learn the names of as soon as possible, because I'll probably ask those on exams at some point. Two examples of fungi: Penicillium chrysogenum. You've probably seen that growing on bread in your refrigerator. If you have some old bread around that's moldy and it turns blue, that's usually Penicillium chrysogenum. That is the fungi from which the original penicillin was isolated. But don't eat Penicillium chrysogenum if you have a bacterial infection, because Penicillium chrysogenum produces other toxins that are very bad for you. So the Penicillium chrysogenum produces some toxins that only kill bacteria, but it also produces toxins that are bad for humans as well. Okay, so Penicillium chrysogenum is a common mold, M-O-L-D, that grows on rotting vegetables and rotting fruit. It was the original source of the antibiotic penicillin. Aspergillus flavus has no redeeming qualities whatsoever. Penicillium chrysogenum is a nuisance. It rots our food, but at least it has a good quality, which is that it produces penicillin for us. Aspergillus flavus has no redeeming qualities whatsoever. There's nothing good about it as far as humans are concerned. It grows on vegetables, particularly corn and peanuts, and if a human eats corn or peanuts or vegetables that are contaminated with Aspergillus flavus, a few years later you will develop liver cancer, because Aspergillus flavus produces toxins, poisonous toxins that are called aflatoxins, and they turn into carcinogens when they transit through the liver. When they pass through the liver, they turn into things that cause cancer. Okay, protists or protozoa, two that we'll learn right away. Plasmodium vivax, which causes a disease called malaria. It is carried by mosquitoes. If a mosquito carries a disease from one person to another or from an animal to a person, we refer to it as a vector. A vector is an insect or some other animal that carries a disease from one person to another. Giardia lamblia causes hiker's diarrhea. It causes a disease called giardiasis. Helminths, two examples of helminths that we've learned already and that you should memorize as soon as possible. Diphyllobothrium latum, that infects sushi and fish if it's not prepared properly. Trichinella spiralis causes trichinosis or trichomoniasis, and it is caught from people who hunt wild animals and then eat the wild animals that they've hunted without cooking the meat properly. You can get a bad case of toniasis. All right, so here we have examples of all of the different types of microbes, examples of viruses, bacteria, yeast, fungi, protists, and helminths. All right, now we've already learned about taxonomy to a certain extent, taxonomy and phylogeny. The term taxonomy refers to the science of grouping things together according to shape and form. The word phylogeny refers to grouping things together according to shape and form and making suggestions about evolutionary relationships. So you put together a bunch of things that have similar characteristics and similar shape and similar form, and then you use that grouping to suggest that they evolved from a common ancestor. And so that's the field of phylogeny. Okay, and then how do we name all living things? Well, a formal way of naming things is referred to as a nomenclature. Right? So a nomenclature is a formal way of naming things. So let's learn about taxonomy, phylogeny, and nomenclature. Right? So taxonomy is where you group things into categories based on common features. Those common features can even include DNA, which is a very powerful way to group things together and prove that they're all related if they have gene sequences that are nearly identical. Okay, so phylogeny is where you group things together based on a presumed evolutionary relationship. Nomenclature is a formal way of naming things, and the way we name things in biology is we use what's known as a binary nomenclature. The word binary means two, and nomenclature means a formal way of naming things. So every living thing on Earth we give two names. One name is the genus name, right? Which genus does a particular organism belong to? And we capitalize the genus. And then the species name is specific to that particular organism. So generally, with large-scale biology, we define a species through a reproductive boundary, which means two animals must belong to different species if they cannot mate and form viable offspring. But if you have any two animals or any two living things that can mate and therefore produce viable offspring, viable means they're healthy and they'll live and they can reproduce and they're fertile. That's what viability means. So if you have two animals that can mate, these two animals look very different and they act very different. If they can mate and produce offspring that survive and reproduce themselves, then they must be members of the same species. Right? So if any two animals or any two living things that can mate and produce viable offspring are members of the same species. Now this gets a little tricky in microbiology because we just said that many microorganisms do not reproduce sexually. So bacteria reproduce by just splitting in half. That's called binary fission. And many protists reproduce by splitting in half, binary fission. And so how do we define a species? So it becomes a little trickier with bacteriology and protistology and so on, but nevertheless we kind of pretend that they have species. The species are generally based on assigning something to one species versus another is generally based on the DNA sequence rather than the ability to reproduce. Okay, so for example, the binary nomenclature of naming Escherichia coli. Look at what we've done here. Escherichia is the genus, and the genus contains several other bacteria that are similar to E. coli, and coli is the specific species. Right? So we capitalize the E in Escherichia, but we do not capitalize the C in coli. And notice that both words are italicized. They're both italicized. So whenever you're referring to a living thing, you have to give both the genus and the species names. The genus name is capitalized, the species name is not, but both words are italicized. Okay, so that's the way you write the name of a living organism if you're a biologist. Okay, so the nomenclature that was developed, phylogeny was developed by an ancient, not an ancient, but a Renaissance scientist named Carolus Linnaeus, and so we use the Linnaean binary nomenclature to give everything a name. And so if you group together living things, you find that all living things have certain things in common. And if you get to the most basic things that are held in common, the most basic thing that's held in common is, does something have a nucleus or not, right? So we call everything that has a nucleus is a member of a domain eukarya, member of the domain. So assigning every living thing to a domain is the most basic degree of similarity between organisms. So any organism that has a nucleus is a member of domain eukarya. If you don't have a nucleus, you're a member of domain bacteria, unless you're a member of domain archaea. So all living things are classified as belonging to one of three domains: domain eukarya, domain bacteria, and domain archaea. And then if you get a little bit more specific, you assign everything to something called a kingdom. And so we have a kingdom Animalia, and we have a plant kingdom, and we have some other things. And to be honest, the term kingdom has fallen into disrepute. It's fallen into disfavor recently. And I'll explain why when we get to the chapter where we're talking about protists. Basically, the biologists have very recently reorganized the top level of the phylogeny tree to include the protists in a different way. So we used to assign things to the animal kingdom and the plant kingdom, and then the protists had their own kingdom. We've erased all that now, and I'll show you what happened with the kingdoms and how we reassigned the location of the protists based on the new phylogeny, which has just taken effect in the last decade or so. Okay, so then you also assign everything to a certain phyla. So every living thing is assigned to a phylum, right? And then every living thing from the phyla, everything is assigned to a class or an order or a family, and then a genus, and then a species. Okay, now, a species is defined, if you're talking about advanced creatures like animals, it's defined by the ability to mate and produce functional offspring, viable offspring. But sometimes with bacteria and viruses, or not viruses, but with bacteria and fungi and a lot of things like that, and even some animals and some insects and whatnot, you do have subcategories within a species. Right? So it's kind of like when the physicists said, let's find the smallest form of matter and we'll call it an atom, and then they decided that the atom is not the smallest form of matter. You have electrons and neutrons and protons, and then they said, oh, that's the smallest thing. Oops, we were wrong again. You can divide electrons into things, and you can divide protons into smaller pieces, and you can have even smaller pieces. Okay, so biologists did the same thing. They started out by saying the most specific way that you can name a living thing is to assign it to a species, right? But then we decided, yeah, but there are different categories within a species as well. So what are we going to call them? What do we call them? So there are two ways that you can describe them. If you have a species, if you have a subcategory within a species, you call it a strain. Right? So a strain is a subgroup within a species. Right? So there are many bacteria that, Escherichia coli is the species name for Escherichia coli. So that should be the end of it. But in fact, it's not the end of it. There are different strains of E. coli that are, some of which are more dangerous than others. And so one of the ways that you can distinguish things at a level beneath, or a level that's even more specific than their species, is to define them by strain. So you have two or three different strains of the same species. Another way that you can describe a subcategory within a species is to refer to it as a serotype. Now the term serotype is very interesting because serotype refers to identifying bacteria or protists or yeasts using antibodies. So there is a science, there's a branch of microbiology and immunology that's referred to as serology. Serology, the sero in serology comes from the word serum, What is serum? Serum is the liquid portion of blood. Right? So serum is the liquid portion of blood, and the liquid portion of blood contains antibodies that are used to kill or destroy invading microorganisms. So the term serology is referred to, means identification of microbes using antibodies. Right? So in this course, we're going to describe many different serotypes of the same species of bacteria, or several different serotypes of the same species of virus. Okay, but generally, genus and species are the two things that we have to know for all of these organisms that we're going to study in this course. Okay, so let's take a look, an example of phylogeny. The panther, the genus name is Panthera, and the species name is Pardus. Okay, so we're going to learn which domain does the panther belong to? Well, clearly it is a eukaryote. Like every cell inside the panther has a nucleus, so it must be a member of the domain. You can tell me it must be a member of the domain Eukarya. Okay, domain Eukarya. All right, it also happens to be a member of the kingdom Animalia, but that term has fallen out of favor in the last few years. Right. Okay, now what about the phylum? It belongs to the panther, along with the humans and any other organism that has a spinal cord, belongs to a phylum called the phylum Chordata. Right, so humans belong to the phylum Chordata, so does the panther. None of the microorganisms that we're going to discuss in this course belong to that phylum because they do not have a spinal cord. Okay, then between the phylum and the genus, we have three other categories called the class, the order, and the family. The panther's class is class Mammalia. The panther's order is order Carnivora, meaning that it eats meat. The class Mammalia means that it has mammary glands. Mammary glands are glands that produce milk. So any animal that produces and feeds milk to its young is classified as a mammal. It is a member of the class Mammalia. And any animal, obviously not all of those animals feed their young, they all feed their young milk, but they don't all eat meat. There are lots of animals like cows, the very obvious example. Cows eat vegetables, they eat grass. Right? And so they are not members of the order Carnivora. The members of the Carnivora eat other animals. Okay, and then the panther is a member of the family Felidae, which is the cat family. So it's a member of the family Felidae. All right, and then the genus and the species names you already know: Panthera pardus. Panthera pardus. Okay, so basically at the very bottom, the only two names that we care about most of the time are the genus name and the species name, which is Panthera pardus. Okay, what about bacteria? Right. So how do you apply this way of naming things to organisms that don't reproduce sexually? Well, it's a bit of a stretch to call bacteria by species names, but we will anyway. Right. So a lot of times the name for bacteria is derived or based on the morphology of the bacteria. So are they cocci, bacilli, or vibrio? That's one thing. Sometimes they're based on the metabolism. And you've already learned that lactobacillus is a bacillus that eats lactose and converts it into lactic acid, which creates acid, and therefore it's an acidophile. So Lactobacillus acidophilus is an example of a bacteria that got its genus name and its species name based not on its reproductive ability, but based on the fact that it's a bacillus and it is also an acidophile. It likes to live in an acidic environment. Okay, protozoans, yeast, and fungi are named the same way through a variety of methods. Viruses are not living things, but viruses they are actually assigned to different families. Viruses are assigned to different families based on what shape they are and what their genome is made out of, whether it's made out of RNA or DNA, and they are assigned to a different family based on proteins that are found in their outer coating. So we'll talk about that when we get to the virology section of this course. Okay, so you can just at some point anytime you like, just take a look at the appendices, Appendix D, and you'll see a lot of these bacteria are listed according to their class, genus, and species. And I just want to draw your attention to this column on the far right-hand side of the table, which tells you various diseases that these things cause. So, Mycobacterium tuberculosis, for instance, causes tuberculosis. Propionibacterium acnes causes acne. These are all members of a domain, the domain bacteria, of course. Okay, these are some more bacteria that are members of a class called Spirochetia, which are also known as spirochetes. They're members of a phylum called the phylum Spirochetes. Spirochetes are squiggly little spring-shaped bacteria. They're kind of shaped like a bed spring. And some of the more important ones that we'll study are Borrelia burgdorferi, which causes Lyme disease. You've already heard about that. And Treponema pallidum, which causes a sexually transmitted disease called syphilis. Okay, some of the viruses, they don't have a domain, but they do belong to an order and a family and so on. So Ebola virus, for instance, right? And then Lissa virus, which causes rabies. Various other things. You can take a look at that appendix as well. They do not belong to a domain. The fungi belong to a phylum, and then they also belong to divisions in genus and species. We're going to be discussing a lot of these different fungi. You've already learned about Histoplasma capsulatum that causes histoplasmosis, for instance. They are members of the domain Eukarya, of course. The amoeba are a type of protist, right? So you've already learned about Entamoeba histolytica, which causes Entamoeba histolytica causes amoebic dysentery. The disease is called enterobiasis, but the common name is dysentery, which is a bloody diarrhea. These are members of domain Eukarya. Right, so we're going to discuss this in greater detail in a couple of lectures, but basically a round bacteria is called a coccus, and plural cocci. Right, so they are round and they're exactly as tall as they are wide. A bacillus, also known as a bacillus singular, bacilli plural, is rod-shaped, has to be usually around 2 times longer than it is wide. There are some bacteria that are classified as coccobacilli, coccobacillus or coccobacilli, where they are only 1.5 times longer than they are wide or less, right? So they're not round, they're not as wide as they are tall. They are slightly bigger in one dimension than the other. But they're not really big enough to be classified as they're not really long enough in one dimension to be classified as a bacillus, so we call those things a coccobacilli. All right, a vibrio is a bacteria that is shaped like a comma, like these things, right? So that's a vibrio. A spirillum is shaped like a spring, but it's usually fairly thick and has 2 or 3 bends at the most. And a spirochete is spring-shaped that is very thin, very flexible, and has many bends, right? So we have coccus, bacillus, coccobacillus, vibrio, spirillum, and spirochete. Those are the basic morphologies. Those are the basic cell shapes of bacteria, and we're going to discuss those in greater detail later on. How are those cells arranged? If they are arranged in a chain, you call it streptobacillus or streptococcus. If they're formed in a cluster, you call it staphylobacillus or staphylococcus. If they occur only in pairs, you call it a diplococcus or a diplobacillus. There are actually no diplobacilli, but there are lots of diplococci. So there are actually lots of streptococci, lots of staphylococci, and lots of diplococci. There's only one example. There are only a few examples of staphylobacillus, and we'll discuss one of those. All right, so Streptococcus pyogenes. If somebody told you that name and you looked at it under a microscope, you would expect that it would be organized in they would be round cells that are organized in chains or strips. Staphylococcus aureus, if somebody gave you that name and you look at it under the microscope, you would expect them to be round shaped but organized in clusters, kind of like bunches of grapes. And Diplococcus pneumoniae, you would expect to see round cells that are organized in just pairs. You know, two round cells together, another two round cells together, and so on. Right, so these are some of the examples of bacteria that we just learned. So two examples of cocci would be Staphylococcus pyogenes and Staphylococcus aureus. You should memorize those as soon as possible. One example of a bacillus is Lactobacillus acidophilus. Another one is Escherichia coli, so you've learned that one as well. An example of a Vibrio is the bacteria called Vibrio cholera, which causes cholera, a disease called cholera. You get it from drinking contaminated water, which causes you to have diarrhea, very bad diarrhea. That's what happens when you have cholera contaminated water. Okay, an example of a spirillum, a good example is called Helicobacter pylori, which we're going to be discussing quite a lot in this course. Helicobacter pylori is an example of a spirillum. And a spirochete, good example of a spirochete, Treponema pallidum, causes syphilis, and we're going to be discussing that a lot in this course. We're going to be discussing Treponema pallidum. So you can memorize the names of these bacteria as soon as you can, because eventually they will start showing up on the weekly quizzes. All right, there are some other features that bacteria typically have that we use to categorize them. Okay, so do they have a flagella or do they have a cilia? There are no bacteria actually that have cilia, but some that have flagella. If they do, how many flagella and how are they arranged? Do they have an outer shell called a capsule? We'll talk about that. Do they produce little seeds called endospores? We'll talk about that as well. These are all additional features that bacteria have that allow us to identify them and to classify them. Okay, so we discussed quite a number of different types of organisms, and we learned the binomial nomenclature for naming them. Okay, let's have a look at what types of microscopes we use to look at things that are so small, biological life forms that are so small that we need a microscope to look at them. So let's look at, for some of you this will be review, especially if you've taken Biology 200, but for everyone else we need to just do a review, another review of the different types of microscopes that are available and what types of techniques you can use to look at different types of microbes. Okay, as I said before, if you take a meter and you divide it into 100, you have a centimeter. If you take a meter and you divide it into 1,000, you have a millimeter. And if you take a meter and you divide it into a million, you have a micron. And a micron is the standard size that the type of scale that you use to look at microbes, at bacteria and protists and so on. If you take a meter and you divide it by a billion, then you have a nanometer, which is the scale that we use to talk about organelles, for instance. So the cell plasma membrane is about 7 nanometers in thickness, and so on. So anything that is, a meter to all the way down to a millimeter or half a millimeter, you can look at with the unaided eye. You don't need a microscope. Right, so these are frog eggs. Frog eggs are individual cells that are just barely big enough to see with the human eye. You can almost see a human egg, a human ovum with the unaided eye, with the human eye as well. It's a little bit small, but you can almost see a human egg without the aid of a microscope. Anything smaller than that, you have to use a microscope. So anything from half a millimeter down to about 0.1 micrometers, you can use a light, you can see with a light microscope, also known as a bright field microscope. A bright field microscope is a microscope where you simply use ordinary visible light to illuminate the specimen. So it's called a bright field microscope because when you're looking at the specimen, you're actually looking down into the light bulb that's attached to the microscope. If you want to go smaller than that, if you want to see things that are down on the nanometer scale, you would have to use a more powerful, more expensive type of a microscope called an electron microscope. So light microscopes use visible light, and there are two types of bright field microscopes, or light microscopes basically. Bright field microscopes can either be a compound microscope or a dissecting microscope. The difference, of course, is that with a compound microscope you slice things up into thin sections, and then you put a light bulb below the slide that you're looking at, and the lens is above the slide, and therefore you're looking at a slice through the middle of the slide. A dissecting microscope is where you look at the surface of things. Like if you're dissecting a dead bird or something like that, you put it onto a dissecting microscope and you look at the surfaces that you're exposing as you cut your way into it. Now, normally with bright field microscopes, you can't, especially with the compound microscope, you can't see any of these small objects unless you stain them, because they're actually so thin that the light travels right through them without stopping, and it's very difficult to see them. So if you want to see something under a compound bright field microscope, you generally have to stain it. So we're going to talk about a few of the different stains that you would use. And so we've become pretty good at this. We've been developing chemical stains for the bright field microscope for centuries now, for a couple of 100 years. And so there are a number of different stains that you can use to stain bacteria and protists and other types of cells in order to see them in a bright field microscope. Okay, but there is a special type of a light microscope, a special type of a bright field microscope, which is called a phase contrast microscope. And there's a variation of the phase contrast microscope called a differential interference contrast microscope, or DIC. And what the phase contrast and what the DIC microscopes do is they use a trick with some light filters that has the effect of increasing the contrast within the cell. So you can sort of almost see individual cells under the bright field microscope if you don't stain them. You can see them very well if you do stain them. But if you don't stain them, if you could find some way to increase the contrast between the inside of the cell and the outside of the cell, between the nucleus and the cytoplasm, if you could find some way to increase the contrast a little bit, you could basically see those cells without having to stain them. And that's what a phase contrast and a DIC microscope do. They have filters that basically, we don't need to go into the details, but they basically just increase the contrast within the cell, which allows you to see things without staining them. Why is that an advantage? Because using the phase contrast microscope, you can look at cells without having to kill them. Normally, if you stain cells, you have to kill them first. But if you're interested in watching cells like bacteria or protists move around, you want to measure how fast they swim, or you want to measure how fast they tumble. Many bacteria move through a type of movement called tumbling. You have to see them in real time, and so you can't kill them and stain them if you want to see them, if you want to measure their speed, or you want to measure the number of times that they move in one direction versus another, which is something that you often would want to look at, right? So you can't do that if you kill the cells in order to stain them. So phase contrast microscopy is a very good alternative that allows you to measure how fast cells are moving and what kinds of things they're doing. Okay, in addition to that, we have fluorescence microscopes. Fluorescence microscopes have a little bit better resolution. Resolution is the smallest detail that you can see. Fluorescence microscopes use the principle of fluorescence. The principle of fluorescence is that the human eye can see certain wavelengths of light. We can generally see wavelengths between around 200 nanometers and 600 nanometers, somewhere in there. If the wavelength of the light is shorter than 200 nanometers, we can't see it, and the light is called ultraviolet. Right? So ultraviolet light, the human eye cannot see it. However, there are some chemicals that will absorb the UV light and at the short wavelength, and then re-emit the UV light at a longer wavelength that we can see. And that's what's known as fluorescence. When you take ultraviolet light and you increase the wavelength so that you can see it, you have a chemical compound that absorbs ultraviolet light and then re-emits the ultraviolet light at a longer wavelength that we can see. So there's a whole lot of microscopy that goes on, microscopic techniques that go on where people are looking at things through the fluorescent microscope. A confocal microscope is where you take two laser beams and you point them at a spot within a cell. And where the two laser beams converge, where they overlap, you will see the cell. That's the only place that you will see any part of the cell. Now what happens is if you raise or lower the convergence point of these two laser beams, you can get an image. You can basically scan up and down within the cell, and in the computer you can take these images that you've generated and put them together into a three-dimensional image of the cell. All right, so you take a cell and you kill it and you stain it with fluorescent stain, and then you take these two laser beams and you focus the laser beams up and down through the cell, and you generate a lot of individual two-dimensional images that are referred to as Z sections, Z or Z planes, right? Z planes. And then you assemble the Z planes or the Z sections in a computer to give you a three-dimensional image of the cell. And that's what confocal microscopy is, also known as confocal laser scanning microscopy. And then finally, electron microscopy uses a beam of electrons instead of a beam of light. Because the beam of electrons has a very short wavelength, you can see a great deal of detail in a very small object. Okay, there are two types of electron microscopy. There are two types of electron microscopes. The transmission electron microscope, or TEM, allows you to see a slice through the specimen, and the scanning electron microscope, or the SEM, allows you to see the surface of the cell. In both cases, you have to stain the cell somehow with metal salts or metal ions, because otherwise the beam of electrons will go straight through the cell and you won't see anything. Okay, so in all of these cases, the specimens have to be stained, so you can't look at live cells using any type of microscopy except the phase contrast microscope. Okay, so what is a resolution limit? So the resolution of a microscope is the smallest detail that you can see, and it's generally proportional to the wavelength of light that you use to illuminate the specimen. Right, so we just mentioned that visible light is light that has wavelengths between 200 and 600 nanometers. 600 nanometers, by the way, is about half of a micrometer, isn't it? So 500 or 600 nanometers is about half a micron, and therefore that means that with light microscopy, the resolution limit, the smallest details that you can see, are about half a micron. If you want to see something that's smaller than half a micron, you have to move to an electron microscope. Fluorescent microscopes, the resolution limit is a little bit better. It might go down to 200 nanometers or 0.2 microns. So it's a little bit better, but not that much. And finally, electron microscopy, you are able to see things on the scale of 1 nanometer. So you can see things down to the size of a single nanometer, which is basically 200 times more powerful than a light microscope. And that's why that's where we get these limits from in the diagram. All right, let's start by looking at bright field microscopy or light microscopy. Right, so we have compound microscopes versus dissecting microscopes. Dissecting microscope is sometimes referred to as a stereoscope. It uses a beam of light that is, the light rays are spread apart or enlarged by glass lenses that refract the light. There are two types of microscopes. The compound microscope is where you slice whatever you're looking at into thin sections, and then you stain it, and then you actually look through it. So the light source is on the opposite side of the lenses. In a dissecting microscope, it doesn't really magnify as much. It uses low magnification, and you're looking at the surface. Okay, so here on the left we have a compound microscope. You put the slide in between the light source and the objective lenses. So the background of the image appears bright because you're actually looking into a light bulb, versus a dissecting microscope on the right, where you simply lay something down on the stage and dissect it, cut it open, and so on, and you look at the surfaces that you're exposing as you're cutting it. So here is a typical thing. This is a section of tissues. This is a section of human tissues that you would look at using the compound microscope. So we're looking at a slice. Notice that it appears to be blue. That's because it's stained. It has been stained. Otherwise, we wouldn't see any of these cells. Okay, here's somebody using a dissecting microscope to cut things open and look at them as they're doing it. Okay, so The magnification limit for light microscopy is about 1,000 times magnification. So that means, if you remember from your first year biology, there's an ocular lens that goes where your eye goes. The ocular lens normally has a magnification of about 10 times. And then the strongest objective lens that you have has a magnification of 100. And the 100 times lens literally has the words oil immersion printed on it. It has the words oil immersion. And that's because when you switch to the 100 times lens, you have to put immersion oil. You have to put a drop of oil onto the slide, and then you lower the lens into the oil, because otherwise the air between the lens and the specimen would diffuse the light rays, and the image would be fuzzy looking. The image would be out of focus. So here's somebody putting a drop of immersion oil on the microscope slide before they rotate the 100 times lens into the oil. So combined with a 100 times lens, objective lens, and a 10 times ocular lens, the maximum magnification that we can get from a bright field microscope is 1,000 times. And 1,000 times magnification is the perfect magnification to use if you're looking at bacteria. That is looking at bacteria is about the smallest things that you can see with a regular bright field microscope. And when you look at them, you still need to use the immersion oil. So if you're going to be a microbiologist, you have to be very familiar with putting immersion oil onto the slides and putting immersion oil onto the lens, and then cleaning the oil off the lens when you're finished, and so on. So here we have a regular lens. If you don't have the immersion oil between the slide, so here's the slide, right? Here's your slide, and here's the objective lens. If there's an air space in between the lens and the cover slip of the slide, the image will be diffused. So to avoid that, we put a drop of oil, and then we lower the lens directly into the oil so that there is no air in between the slide and the lens. And that allows the light rays to avoid being scattered by the air, and then you can see things at 1,000 times magnification. So here we have bacteria. These bacteria look nice and sharp. You might remember from first year biology that when you looked at bacteria, a lot of you had trouble focusing the image. A lot of you might have had trouble focusing the image if you were looking at bacteria, and it was probably because the immersion oil wasn't touching the lens or something like that. And some of you may have noticed that you had trouble focusing the 40 times lens, never mind the 100 times lens. You had trouble focusing the 40 times lens, and everything looked blurry and fuzzy. And the reason for that might very well have been that the person who used the microscope before you put oil onto the wrong lens. Instead of putting the oil onto the 100 times lens, they accidentally put the oil onto the 40 times lens, and then they didn't clean it off. So that would lead to you're not being able to focus something using the 40 times lens or the 10 times lens. Okay, now depending on what you're looking at under the compound microscope, you have to prepare the sample, right? So if you're looking at tissues, normally what happens is you embed the tissues into paraffin wax, and then you slice the tissues into thin slices using a machine called a microtome, and then you stain them. This is common for the science of histology. The science of histology is looking at tissues like skin and liver cells, and cutting up the liver and looking at a slice of the liver, cutting up a kidney and looking at a slice of a kidney. So you embed the things in paraffin wax. That's to avoid having the tissue collapse when you cut it with the microtome, and then you stain it. Now, if you're talking about bacteria, which is mostly what we're going to be talking about in this course, you smear it instead of slicing it. Right? So you take a bacterial petri plate, for example, and you have a lump of bacterial cells called a bacterial colony, and you take a little metal loop, and you scoop some of those cells off of the colony. You did this in Biology 110. You scoop some of those cells off, and then you smear them onto a glass slide. And then you fix them onto the glass slide, usually by heating it from below using a spirit lamp. And that basically fries the bacteria onto the glass so that they won't wash off when you try to stain them. Right? So you smear the bacteria onto the slide, and then you fix them onto the slide by frying them onto the glass using heat. and then you stain them. Usually you stain bacteria with a series of stains that are referred to as the gram staining protocol. And the gram staining protocol is useful because, if you remember from first year, it allows you to tell the difference between a gram-positive bacteria and a gram-negative bacteria. So the gram staining protocol is an example of a differential staining protocol because it allows you to differentiate or to tell the difference between two types of bacteria. If a bacteria has a peptidoglycan cell wall, it will absorb the crystal violet gram stain and it will appear purple. If it doesn't have a peptidoglycan cell wall, it will stain pink when you counterstain with safranin. Okay, so let's look at making a smear of bacteria. Okay, so you take a wire loop like this one, right? And you put it first. You start by putting a drop of water onto the slide because otherwise the bacteria will end up being smeared too thickly onto the slide. And then you take a metal loop, we call that a loop, and you scoop up some of the bacteria off of the Petri plate that you were working with, and then you mix them in with the water and you smear it around until the bacteria are nicely separated. And then you pass the slide a few times through a spirit lamp, through the flame of a Bunsen burner. If you heat it too much, you'll burn them all to a crisp and you won't see anything. If you don't heat it enough, they'll come off when you try to gram stain them. So it's kind of an art. It's not necessarily a science as much as it is an art to get to heat the slide just enough to fix the bacteria to the glass slide. Okay, so you do heat fixation by passing the glass slide gently and quickly a couple of times through the spirit lamp, the flame of a Bunsen burner or a spirit lamp, and knowing exactly how long to leave it in the flame is something that you can only learn by practicing it. We won't have a chance to practice it in this class because we don't have a lab for this class, but you did do a little bit of it in Biology 110. Okay, then you dip the glass slide in various types of stain, or you pour the stain onto the glass slide in order to stain what's on the slide. Okay, so here we have some bacteria that have been stained. Okay, I don't know if you remember this from the first lecture or in the first part of this lecture, but notice that we have some cells. How would you describe the morphology of these cells? If you want to stop the recording and try and guess, you can. Okay, and I'll tell you the answer. So these cells are longer than they are wide. They're more than 2.5 times longer than they are wide, so therefore these are bacilli. These are classic rods. These are classic bacilli. And not only that, but these bacilli are attached together end to end. That's a function that's caused by the way they divide. So when these particular bacteria divide, they divide end to end rather than one on top of the other, and they end up staying together after they've divided. And so we would describe that as a streptobacillus. Streptobacillus because you have bacilli that are in chains, and this particular streptobacillus is Bacillus anthrax, which causes a disease called anthrax. Bacillus anthracis causes a disease called anthrax. Okay, if you slice things, there's a slightly different protocol. Right, so first you take a piece of tissue and then you embed it in paraffin wax. Paraffin wax, if you've ever made jam or something like that, you know that you preserve the jam by melting this wax and then pouring it on top so that you've sealed the jar, and then you put the lid on. So in this case, we are pouring paraffin wax onto the specimen to give it strength. And then we take this chunk of tissue out of the mold and we cut it up in a machine called a microtome. I don't know if you've ever been to a delicatessen where they slice meat in a meat slicer. A microtome is exactly like a meat slicer, only it slices much finer slices. So it slices slices of this embedded material. It slices it into 1 micron thicknesses, 1 or 2 microns, depending on how thinly you've set it to slice. And then those slices are thin enough that you can stain them and then see them in the microscope. Okay, so you slice the embedded tissue that's embedded in paraffin into thin slices. If we had not embedded the tissue in paraffin, when the blade of the microtome tried to cut this block of tissue, it just would have crushed it. Right, so you've seen that happen when you try to cut. If you try to cut a loaf of bread with a knife and the knife is kind of dull, instead of cutting the bread, it just crushes it. right? And so that's what we're trying to avoid here. So if we hadn't embedded the tissues in paraffin, the microtome just would have crushed it instead of cutting it. So then we put these little slices onto slides, and then we do what's called chemical fixation. We dip them in several different types of alcohols in order to make them kind of dry out and stick to the slide. And then we dip them into the staining jars. These jars are called Copland jars. You don't need to know that, but that's what they're called. And then when you look at them under the microscope, you have these tissues that are composed of many different cells, and they are beautifully stained. All right, now let's move on to phase contrast microscopy. Phase contrast microscopy is where we use a trick with a light filter to increase the contrast between different structures within the cell so that we don't need to stain it. Okay, so if we use a filter to exaggerate the differences in contrast, then we don't have to kill the cell in order to see it. So this is the only technique where we don't actually have to kill the specimen. So this is useful for studying microbial motion, or locomotion and feeding behavior, watching bacteria move around and eat things, watching protists move around and eat things. You can recognize a phase contrast image because it kind of has the cells kind of look like they have a halo around them. They look like they're luminescent or glowing. And then there's an extreme version, a very extreme version of differential interference contrast, phase contrast microscopy that's called DIC. DIC stands for differential interference contrast, which is an extreme version of phase contrast microscopy. And I'll show you what each of those look like. Okay, so here we just take an ordinary light microscope, and we insert two different filters above and below the specimen that have the effect of increasing the contrast inside the specimen that we're looking at. Okay, so this is what you would see if you looked at human cells without staining them at all. You can barely see them. This is what they look like if you stain them. So they're much more clear and much more easy to see if you stain them. Okay, but what if we don't stain them? What if we use phase contrast instead? This is what they look like using phase contrast. And notice they look like they're kind of glowing, or they're kind of look like they're luminescent. That is a classic telltale sign. That's a giveaway that you're looking at these cells through a phase contrast microscope. Okay, so here we have a paramecium, which is one of the protists that we'll study in this course. These things live in the water, and they dart around the water at a very high speed, and they eat things. And sometimes you want to measure how many things they eat in a minute, and you can actually do that in real time by counting the number of things that they eat. But obviously you couldn't do that if you had to kill them and stain them. So it's lucky that we can use phase contrast microscopy to measure the behavior and the movement of these organisms in real time. Okay, so now differential interference contrast. Okay, so here we have unstained human cells. These are buccal cells, by the way. Any of you that were in my anatomy class should remember where those come from, right? So buccal cells come from the inside of your cheek, and they're easy to dislodge. You just rub a Q-tip, or you rub a swab or a toothpick on the inside of your cheek, and then you smear it onto a microscope slide, and you have buccal cells. Okay, so buccal cells, these are what buccal cells look like if they are not stained. This is what they look like if you view them using phase contrast. And then if you look at them using differential interference contrast microscopy, they look like this, right? So these are two very distinctive looks for two types of contrast microscopy. All right, let's talk about the fluorescence microscope for a minute. Okay, so visible light is wavelengths of around 400 to 600 nanometers. UV wavelength begins at about 400 and goes all the way down to 200 or lower. And so in this case, you have a regular microscope, and you just substitute an ultraviolet light source. So here we have a fluorescent microscope. Here we have the objective lenses. You're used to those. Here we have, those are the ocular lenses. These are the objective lenses down here. Here's the stage where you put the slide. And then underneath here, there's a regular light source. But back here, there's an ultraviolet light source. Right, so this ultraviolet light source back here is refracted through a prism, and it's basically refracted down onto the specimen through a prism that's located here. So this fluorescence microscope costs about 10 times as much as a regular microscope. A regular microscope that we use in the first year lab is about $1,000. It's not cheap, but this is considerably less cheap. It's about well, this I'm not 100 times, but 10 times. Sorry. So a typical fluorescent microscope costs about $10,000. Right, and in order to see the specimen, you have to stain it with fluorescent stains, and so there are fluorescent dyes that will fluoresce green or red once they're exposed to ultraviolet light. And so once you get the image, they kind of look like neon signs, right? So they look a little bit like neon Coca-Cola signs, because you see a fluorescent image on a black background, because the background is black because we've turned off the regular light, and now we're illuminating the specimen with an ultraviolet light that is bounced off the prism that's above it. And so the specimen is fluorescing because the UV light is being absorbed by the ultraviolet light is being absorbed by dyes and then re-emitted at a longer wavelength. All right, confocal microscopy. This is where you take two different laser beams and you point them at a common spot in the cell. The cell will only be visible at the spot where the two laser beams converge. So you can cause this convergence point to go up or down within the cell, and you generate a lot of things that are called Z sections or Z planes that look like slices through the specimen. And then you take a computer which collapses the Z planes or the Z sections. That's the official word for that's what they call it. They say you collapse a bunch of Z planes and then you make a three-dimensional image inside the computer. So here we have a bunch of Z planes that were generated by two laser beams focusing upwards or downwards within a cell. Right, so you can see the image number 1 at the top left is right at the top of the cell, and then we go down, 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 down. It gets bigger and bigger because we're getting towards the middle of the cell, and then it starts to get small again because we're getting to the bottom of the cell. And then the computer regenerates a three-dimensional image, like so. Right, so that image was generated by a confocal laser scanning microscope to give you a three-dimensional image that, because it's generated in the computer, you can rotate it around. All right, let's look at electron microscopy. So if you use a beam of electrons instead of a beam of light, you can get much greater detail. Generally, the images are black and white because the electron beam always doesn't take images in color. And the beam, instead of being spread out by glass lenses the way light microscopes are, the beams of electrons are spread out using copper magnets that are formed into rings around the electron beam. So here's an electron microscope. It's very expensive, a couple $100,000 easily. It uses high voltage. It's very expensive and a bit dangerous because of the high voltage, so you have to be trained to use it properly. Usually a university buys a few of these microscopes and they allow the individual professors to use them if they pay them rent. So if you end up as a graduate student in a university who's doing research with the electron microscope, your professor would have to pay your department a certain amount of money per hour to let you use the electron microscope, and if you break it, it'll be a really big scandal. Okay, so two types of electron microscopes. The transmission electron microscope sees slices of things. The scanning electron microscope sees surfaces. Right, so TEM on the left, SEM on the right. Okay, so TEM looks at surfaces, TEM looks at slices through things, and in order to see things you would have to stain them with metal salts like osmium tetroxide. And the SEM sees surfaces, but it will not see the surface unless you coat the surface with vaporized metal first. So you would normally coat the surface of cells or whatever it is you're looking at by spraying it with molten vaporized gold or molten vaporized silver or lead. Okay, so that flea, it's not really microscopic. You can see a flea with the naked eye, but it's been sprayed here with gold in order to see it under the scanning electron microscope. Here's a spider, the head of a spider. Usually a spider's head is big enough that you can see it without a microscope, but we can see a lot of detail if we look at it using the scanning electron microscope, and yes, that means somebody had to spray paint a spider with liquefied gold. So you see in laboratories that have a lot of electron microscopy work, you'll see all sorts of things sprayed with gold that are unusual. You know, I once had a friend who worked in an electro. She was a graduate student, and she was a little bit strange, and she liked ugly, hideous things, and so she took one of these gold-plated spiders and made it into a brooch. I don't even like to think about it. Okay, so false color SEM simply, or TEM, false color electron microscope images simply means that you take the black and white images and you colorize them using Adobe Photoshop, or you could even use water paint or crayons if you wanted. And it's not done to be deceptive. Everyone knows that the images are generated in black and white, and they've just colorized it to make it easier for you to distinguish one part from another. So here we have red blood cells, and in between the red blood cells we have a trypanosome, Trypanosoma cruzi. And it's kind of nice to paint the blood cells red so you can tell that those are red blood cells, and then you paint the Trypanosoma cruzi a yellowish color. I don't know why, but that obviously makes it easy to spot them in between all of these red blood cells. So we are going to be talking a lot about Trypanosoma cruzi in this course. Okay, so we talked about several different types of microscopy and about the resolution limits for the different types of microscopes, and we also talked about that in context with the sizes of the cells that we're going to be looking at. Okay, now how do we stain things? We talked about how critical it is to be able to stain things so you can see them, and there are several different types of stains that are used by microbiologists, and so we need to become familiar with some of those. Okay, so remember, you won't see anything under the microscope unless you stain it under the bright field microscope. Okay, so there are generally two types of stains. Number one is called a general stain, or an oversight stain, which stains everything more or less equally. Another type of stain is called a differential stain, which stains things selectively. So it stains one type of macromolecule but not another. It might stain, for instance, there are stains that are positively charged chemical stains that will stain negatively charged nucleic acids. Remember that at neutral pH, at pH 7, DNA has a negative charge. So if you can find a stain that is positively charged, it will selectively stain the DNA in the nucleus of a cell. On the other hand, proteins tend to have a net positive charge. A lot of proteins have a net positive charge, and so if you have a stain that has a negative charge, it will selectively stick to the proteins in the cytoplasm of a cell, but it will definitely not stick to the DNA in the nucleus. So those are considered to be differential stains. They allow you to differentiate or tell the difference between one organelle versus another. Okay, counterstaining is what you do if you do differential staining that stains one thing, and then you want to see everything else in the cell, right? So sometimes it doesn't do you any good to see the nucleus unless you can see the rest of the cell as well. You want to stain the nucleus in one color and the rest of the cell in another. So generally what you can do is you can stain one part of a cell, or you can stain one organelle with a differential stain, and then you stain everything else with a counter stain. All right, so a mordant is a chemical that allows any of these stains to stick, right? So sometimes if you stain parts of a cell, the dye will be preferentially absorbed by one organelle but not another. That's fine. But that dye will wash away because it's not stuck to it very strongly. That dye, it may have stuck differentially or preferentially to one organelle and not another, but it can easily be washed away, so that's no use to you. So sometimes you treat cells that have been stained with something called a mordant, and a mordant is a chemical that forms a covalent bond between a specific stain and whatever it is that that stain is sticking to. So in the Gram staining protocol, which we're going to learn soon, the mordant that's used is iodine. Some of you know that already. So, but iodine is just one example of a mordant. So in general, a mordant is a chemical that specifically forms covalent bonds between the dye that you're using and the target of that dye. Okay, so examples of general or oversight stains. Right, right, so safranin is a general stain which will stain everything equally. So here you see saffron on the right. That's what's staining these paramecia. And neutral red is another general stain. So I might, in a quiz, I might ask you, tell me the names of two general stains. Safranin is one, neutral red is another. Two general oversight stains, safranin and neutral red. So you should definitely remember those names. Okay, examples of differential staining. They mix hematoxylin and eosin together into a compound dye called H&E stain. And H&E stain is used to stain tissues so that you can see the nuclei. The nuclei are a blue or purplish color, and then the cytoplasms are pink. And so H&E stain stands for hematoxylin and eosin, which is a common histology stain. Okay, so here's methylene blue. We have a bunch of cells that are in a device called a hemocytometer. A hemocytometer is a little tiny microscope slide that has a grid etched into it, right? So somebody took a drill and they etched this grid into the slide, which makes it easier to count the numbers of cells inside each of these smaller squares. So a hemocytometer is actually used to count the number of cells that you have per mil. And in this case, if you stain the cells with methylene blue, you can not only count the number of cells per milliliter, but you can also count the number of live versus dead cells per milliliter. So you would typically count 5 or so of these cells, and then you would then count up the number of live cells and the number of dead cells in the hemocytometer, and then the methylene blue would allow you to tell the difference between live versus dead cells. Okay, these purple cocci, these dark blue or purple cocci have been stained with crystal violet. So these are gram-positive cocci. This is a histology section, so somebody has cut a slice of tissue. This might be human tissue or animal tissue, and you can easily see the nuclei because they're stained dark blue or purplish color. That's because of the hematoxylin. And then the cytoplasms of all of these cells, as well as connective tissue. This is all connective tissue that doesn't contain cells, but it's made of proteins, will stain pink. All right, the gram staining protocol, it's critical that we learn all about this for this course because this course has a lot to do with bacteria. Okay, so the gram staining protocol allows you to divide all bacteria into two different categories, gram-positive or gram-negative. Gram-positive cells have a peptidoglycan layer on the outside, whereas gram-negative cells don't. The crystal violet dye stains the peptidoglycan dark purple. And then after you've done that, you counterstain the cells with safranin. So if the cells had a peptidoglycan cell wall, they will appear purple when you look at them under the microscope. If they didn't have a peptidoglycan cell wall, they will appear pink because you counterstained them with safranin. Okay, so gram-positive bacteria appear purple after the gram staining protocol, and gram-negative bacteria appear pink after the gram staining protocol. So you can see that these cocci are gram-positive cocci, and these rods are gram-negative rods, gram-negative bacilli. So a protocol, an experimental protocol, is basically a recipe. Imagine that you were a cook and you were trying to figure out how to cook a certain dish, how to prepare a certain dish. You would look it up in a cookbook. Well, when you start working in the lab, you'll see that there are protocol books on all the bookshelves. And those protocol books tell you which chemicals to mix together and in which order and how to do certain experimental techniques. So this is an example of an experimental protocol called the gram staining protocol. And what you do is it allows you to distinguish gram-positive bacteria from gram-negative bacteria. That is the number one way that we classify bacteria. If you have an unknown bacteria, the first question that any microbiologist would ask is it gram-negative or is it gram-positive? And then if you say, well, it's gram-negative, after that they would ask, well, does it have this or that, or what types of genes does it have, and so on. But the first question will always be, is it a gram-positive or a gram-negative bacteria? Okay, so what do you do in the gram stain protocol? You smear the cells onto a slide and you fix them using a spirit lamp like I showed you in that earlier slide. You use heat fixation. Right? And then the first step is you stain with crystal violet. So you take a dropper and you drop crystal violet onto it. You leave it for about a minute. And then you shake the crystal violet dye off to get rid of the excess. Now at this point, the crystal violet will have stained both gram-positive and gram-negative bacteria, but the stain is not sticking strongly to the gram-negative bacteria. We then add a mordant. In this case, the mordant is iodine, which forms a covalent bond between the crystal violet and the peptidoglycan. And then we rinse the cells with ethanol, which causes the crystal violet to be washed away from the gram-negative cells. And then we counterstain any gram-negative cells with safranin. And at the end, gram-positive cells look purple, gram-negative cells look pink. So here we are. We take a dropper and we drop some crystal violet onto the cells. We leave it for a minute or so. Then we add some iodine in order to form a covalent bond between the peptidoglycan and the crystal violet. But look at this point, at the beginning, all of the cells appear to be dark purple because we haven't actually washed away the crystal violet yet. Okay, then once we have fixed or permanently attached the crystal violet to the gram-positive cells with the iodine. Then we decolorize. We rinse with alcohol, and this has the effect of rinsing the crystal violet away from any gram-negative cells. And then we counterstain with safranin, which causes the gram-negative cells to turn pink. And so there you have it. We can see, using the gram staining protocol, we can see that this specimen, this sample, contains gram-positive cocci and gram-negative bacilli. Let's look at a few other identification and staining techniques, and you do need to memorize these. You need to memorize the gram staining protocol. You need to memorize what each of the dyes and what the mordant does and what the purpose of the alcohol and the iodine are. And now let's look at some other techniques that are used to identify bacteria and yeast and a few other things. All right, first, memorize this. You need to know this. There's a chemical called carbylfusion that is used for a technique called acid-fast staining. Acid-fast staining is where you take the chemical carbylfusion and you use it to stain cells that have something called mycolic acid in their cell wall. Okay, so there are a bunch of there are a bunch of bacteria. There are several different types of bacteria that contain a compound called mycolic acid in their cell wall. And the mycolic acid makes it difficult to stain with gram stain. Right? So, but you can still stain them with gram stain, but you also want to know whether they have mycolic acid in their cell wall, because many of the bacteria that have mycolic acid in their cell wall are dangerous. Example, Mycobacterium tuberculosis. You should memorize the name Mycobacterium tuberculosis, because this is the name of the bacteria that causes tuberculosis, which is a respiratory disease. It's where you get a bacterial infection of the lungs, which is potentially quite lethal. If untreated, tuberculosis, if you don't treat people who have tuberculosis, around 40% of them would die. So these days, if somebody is diagnosed with Mycobacterium tuberculosis, they go into the hospital and they are given antibiotics and they get better. But we have to have some way to determine whether or not they're infected with Mycobacterium tuberculosis, because there is a specific antibiotic that will kill Mycobacterium tuberculosis, and there are other antibiotics that don't. Right? So there are many antibiotics that are specific, that will specifically kill one type of bacteria but not others. So there are specific antibiotics that we have to use to cure somebody of Mycobacterium tuberculosis or tuberculosis. But first we have to identify or prove that they are actually infected with Mycobacterium tuberculosis. And so how do we do that? We use acid-fast staining. We use carbylfusion. Right? So carbylfusion is used for acid-fast staining. Acid-fast staining is used to identify any bacteria that has mycolic acid in their cell wall. And an example of a bacteria that has mycolic acid in the cell wall is Mycobacterium tuberculosis. You can see myco in the name of the bacterium. Myco, as the term mycolic acid is derived from Mycobacterium. Okay, so there's another one which is Mycobacterium leprae, and that causes the disease called leprosy. And so if you're gonna diagnose somebody, if you're a doctor, somebody is sick and you want to diagnose them as having, if you want to determine whether or not they have tuberculosis, you would take a sample, you would get them to spit up into a cup, that's called sputum. And then you would take that sputum and you would smear it onto a slide, and then you would stain it with carbofuscin, and if it stains it, then they have tuberculosis. They have Mycobacterium tuberculosis. So this is a diagnostic tool. In this case, carbofuscin, acid-fast staining, is a diagnostic tool, so you can figure out which bacteria somebody is infected with in order to cure them. And this is what the bacteria look like after they've been stained with carbofuscin. Fuchsia is kind of an orangey color, and so this carbofuscin stains these bacteria a kind of a reddish orange color. All right, now let's look at something else. There are some bacteria and there are some fungi and some yeast that have an outer structure called a capsule. Right? And so again, if you want to prove that somebody is infected with a specific type of bacteria or a specific type of yeast, you can look to see if whatever microbe is infecting them has a capsule. Now, a capsule is is a type of a polysaccharide shell, and there are two types of polysaccharide shells, but one is called a capsule, one is called a slime layer, and we'll talk about that later. But both a capsule and a slime layer are both examples of a structure called a glycocalyx. Right, so we're going to talk about glycocalyxes later. But a glycocalyx is a layer of polysaccharide that you find outside of the cell, and it actually prevents gram staining from getting into the bacteria, so it's very hard to analyze the bacteria sometimes. But what you can do is you can kind of counterstain the liquid that the bacteria is floating in, and if you see this kind of an empty space where the stain didn't stain, then you assume that there's a bacteria or a yeast that has a glycocalyx inside that halo, that area that's not staining. So there are several examples of bacteria that have a glycocalyx. There's one example of a yeast that has a glycocalyx, and it's called Cryptococcus neoformans. Cryptococcus neoformans causes a disease called cryptococcal meningitis, and we'll talk about that later. But one of the ways that you can counterstain to visualize a capsule is with India ink. Just simple old ink, regular old ink. Okay, so here on the left, you see the ink has stained the liquid that these yeast cells are floating in, but it's missing, the stain is not able to penetrate the capsule, so these hollow spaces that you see are actually the cells that are surrounded, the Cryptococcus neoformans cells that are surrounded by a capsule. Okay, now there are some bacteria, usually some of them are quite dangerous, that produce little seeds. Seed is a bad word, but they produce little tiny seed-like things called endospores. Of course, the word endo means inside, and the word spore specifically refers to haploid germline cells. So you know that angiosperm plants produce seeds, and a seed is actually a diploid thing. Right, so a seed is basically a seed contains a plant embryo, which is diploid. But you know that there are other plants like ferns that do not produce seeds, they produce spores. And the difference between a seed and a spore is that a seed contains a diploid embryo, and a spore is a haploid germ cell, so it only contains one copy of the genome. Okay, so we already learned that bacteria are haploid. Bacteria are never diploid. So when they produce a seed, it's not going to be a seed, it's going to be a spore. Right? So the spore contains the chromosome of a bacteria. And so there are certain bacteria that when they are starving, if you starve them for food, they basically say, well, I'm going to die, but at least my children will survive. So they produce a spore, an endospore. The cells die, but the spore survives, and then the spore will hatch or germinate if the spore lands on fertile soil or food, is an area where there's food. So there are only some bacteria that make an endospore while others don't. So it's to our advantage. We can use the presence of an endospore as a diagnostic tool. It's to our advantage to be able to specifically see whether a bacteria is making an endospore or not. And then if we see that it is, then we can say, oh, well, it must be this bacteria or that bacteria. Example, Bacillus anthracis, which causes a disease called anthrax, produces endospores. Okay, now there's a specific stain that you use to stain endospores, and it is called malachite green. So you do need to memorize malachite green. You need to memorize the name malachite green because it is a commonly used stain that is used to identify bacteria like Bacillus anthracis. Okay, so you can see here that they have stained these Bacillus anthracis cells. Bacillus anthracis is a bacillus, like the name says. And I mentioned in an earlier slide that Bacillus anthracis forms strips, so it's a streptobacillus. You see the little green things in the middle? Those little green things are endospores, Bacillus anthracis endospores. And in order to see the rest of the cell, they counterstained the rest of the cell with safranin. So here we stained the spores with malachite green, and then we counterstained with safranin so you can see the endospore inside the cell. All right, now we mentioned the fact that many bacteria have a flagellum, singular, or several flagella, and that we classify or identify bacteria based on whether it has a flagellum or not. And if it does have a flagellum, does it have just one? Does it have more than one? If it has more than one, how are they arranged? Are they all on one side? Are they completely surrounding the entire cell? However, the flagella are usually so thin that you can't see them under the microscope, and so you have to add a thickener so that you can visualize them. Right? So the thickener that we use is a dye called basic fuchsin. Right? So don't confuse basic fuchsin with carbol fuchsin, which is what we were using to stain mycobacteria. Basic fuchsin is a stain that is generally used to stain flagella. It doesn't do anything to identify bacteria that have mycolic acid. Okay, so basic fuchsin is it's a thickener that kind of coats the flagellum and then allows you to see a flagellum much more easily. Example, Helicobacter pylori is what's known as a lophotricious bacterium because it has a whole bunch of flagella, but only at one end. It's a bacillus. It's actually a spirillum, but so it's kind of shaped like a spring, but it has all these flagella at one end. So here we're looking at a bacteria called Bacillus cereus, which is a common contaminant of steamed rice. So if you ever got food poisoning from steamed rice, it was probably the result of Bacillus cereus. So we're going to talk about Bacillus cereus in this course later on. And you can see that if you stain Bacillus cereus with basic fuchsin, it thickens up the flagella so that you can see these cells are completely covered with flagellum, and that's an example of a paratricious bacteria. Paratricious meaning that the entire outside of the cell is covered with flagellum. Okay, so to summarize the different stains that we've learned about, we learned about the Gram stain and all of the components: crystal violet, safranin, using safranin as a counterstain, using iodine as a mordant. We learned about acid-fast staining using carbol fuchsin. We learned about capsule staining using India ink. We learned about endospore staining using malachite green, and we learned about flagellar staining using basic fuchsin. You need to memorize all of those things as soon as you can, because those names of stains will start to appear on the quizzes fairly quickly. Okay, so what about fluorescent microscopy? All of those staining techniques that I just explained to you only apply to light microscopy. In fluorescent microscopy, we simply use fluorescent dyes that absorb ultraviolet radiation and re-emit it at a longer wavelength that we can see. So the visible light spectrum is between around 400 and 700 nanometers. Notice that if you take light wavelengths that are shorter than 400, they are called ultraviolet light, way down here somewhere. And our eye cannot see those because they're below, the wavelength is too short for us to see. Okay, so we use fluorescent dyes. Now, we're not really going to get into this in this course, but there are two dyes. The two first dyes that were ever invented for fluorescent microscopy were called fluorescein, which has a green color when it fluoresces, and Texas red, which has a red color when it fluoresces. And each of those two dyes has what's known as an absorption spectrum and an emission spectrum. So fluorescein absorbs ultraviolet light at a wavelength of 495 nanometers, and you'll notice that when you looked at that chart that I just showed you, 495 nanometers is not completely ultraviolet. Some of it you can see. But what it does is it re-emits, it absorbs light at 495 nanometers, and it re-emits it at 519 nanometers, which has a green appearance to the human eye. Texas red absorbs at 589, which is barely ultraviolet at all, and re-emits at a wavelength of around 615 nanometers, which is red in color to our eye. So if you go on in science and you do a lot of microscopy work with the fluorescent microscope, there are all kinds of tricks that you can play to get different parts of the cells to fluoresce based on which wavelength of light you use to excite the fluorescent dyes. So if you use 495, then you'll get preferentially the green light coming from fluorescein, and if you use 598, you'll get preferentially the red light coming from the Texas red. So you can kind of if you're a real dedicated fluorescent microscopy expert, you can do all kinds of tricks by manipulating the different fluorescent dyes that you use, and also by manipulating the different wavelengths of light that you use to excite the dyes. But most of the time, if you're just doing regular common fluorescent microscopy, you're looking at reds and greens. Okay, so the electron microscope would go right through living tissue and you wouldn't see anything, so you have to stain it, and usually with metals. Okay, so we stain slices of tissues for the transmission electron microscope with a metal dye called osmium tetroxide. You do need to know that for the tests. And for scanning electron microscopy, we coat the surfaces of the specimens with gold or silver. So you do need to know that as well. All right, now there's a very specific example of staining. Okay, so we just heard, for instance, that hematoxylin is a negatively charged dye that will preferentially stick to DNA, because it's a positively charged dye that will stick to negatively charged DNA. And then we learned that eosin is a pink dye that is negatively charged that will preferentially stick to positively charged proteins. Okay, but what if we're interested in one specific protein? If we try to stain a cell with just eosin, we'll stain all the proteins at once, which is no use to us. We want to be able to stain one specific protein. Okay, if we're going to do that, we can take advantage of the immune system. We can take advantage of the animal immune system by what we can do. If you remember from first year, you remember that the immune system produces antibodies. Those antibodies will stick to one specific protein from a foreign invader, one specific foreign invading protein. Okay, so what if we make antibodies to a specific protein, and then we label the antibodies with either with an enzyme or with a fluorescent dye? Then that will allow us to visualize or see just one specific protein in a cell, and then we can see where that protein is. That technique is referred to as immunolabeling. And so what you do is you purify a specific protein from an organism that you're interested in, like a bacterium, right? So you purify a protein using biochemistry techniques, column chromatography or something like that, and then you inject it into a rabbit or some other animal, And the rabbit's immune system will make antibodies that stick specifically to that protein. And then you take those antibodies out of the rabbit's blood and you label them with either an enzyme or with a fluorescent dye. And then you put these labeled antibodies onto the slide. They will stick only to that one specific protein and to no other protein. So that is an example of a very differential stain, a very specific stain. Okay, so in bright field microscopy, we stain the antibodies with an enzyme that causes a color change to a chemical. In fluorescent microscopy, we simply stain the antibodies with fluorescent dyes. And in electron microscopy, we attach microscopic gold beads to the antibodies. Microscopic gold bead particles, microscopic gold beads are referred to as colloidal gold particles. And they will stick naturally to antibodies. And so these are the three ways that we label antibodies if we want to visualize a specific protein using either bright field microscopy, fluorescence microscopy, or electron microscopy. Okay, so let's say we're doing this with bright field microscopy. We purify a protein, let's call it protein X, just because I can't think of a better name. And we purify protein X from a bacteria. We inject it into a rabbit. The rabbit will make anti-X antibodies that will attach specifically to that protein, to protein X. And then we take an enzyme called horseradish peroxidase that will turn a chemical called DAB. It will turn it brown color if it's there. And so that enzyme is now attached to the antibody. We call that conjugation to an antibody. And then you put the labeled antibodies onto a slide, and anywhere that protein X is, you will see a brown color. And then you counterstain the rest of the cell so you can see where the rest of the cell is. Right? So we purify protein X, we inject it into a rabbit or a rat. The rabbit makes antibodies that will stick only to protein X. Then we conjugate horseradish peroxidase, HRP, which is an enzyme that turns a chemical called DAB brown. We put that onto the slide. That means that anywhere that you find protein X on the slide, it will turn brown. And then we counterstain using a stain called trypan blue, and that this allows you to tell where in the cell that specific protein is. Fluorescence microscopy. Okay, you tell me what the protocol is. You can stop the recording and see if you can tell it to me, because I might ask you this on a test. All right, so I'll tell you the answer now, of course. You purify protein X, you inject it into a rabbit, you take the antibodies, and this time you simply stain the antibodies with fluorescein, for instance, because fluorescein will naturally stick to proteins, and these antibodies are made of proteins as well. So you will stain the antibody, which is a protein, and then the antibody will stick to protein X, and then you'll be able to see where it is. And then you simply counterstain the rest of the cell with Texas red, for example. Right, so here you purify protein X, you inject it into a rabbit, you take the antibodies, you attach fluorescein, FITC fluorescein, to the antibodies, so the antibodies are now fluorescent green, and then now these antibodies, you can see where they went. So this protein is localized around the nucleus, and then you counterstain the rest of the cell with just counterstaining with Texas red. The electron microscope. Okay, so this technique is really usually used just for transmission electron microscope, but you attach microscopic gold beads called colloidal gold particles to the antibodies that you've created, and then you put them on the slide, and then you counterstain the rest of the cell, counterstain the rest of the slide with osmium tetroxide. All right, so here we've created an antibody which is going to stick to the antigen, protein X in this case, and we have conjugated or stuck colloidal gold beads onto the antibodies. So here we can see where these dark spots are where the antibodies have stuck, and all the other area is counterstained with osmium tetroxide. All right, now here's another interesting type of experiment that you can do with a fluorescent microscope. You can do something called a colocalization experiment. The word colocalization means proving that two things or two proteins are in the same place at the same time in the cell. Why would you be interested in doing that? Well, one of the reasons you'd be interested in doing that is because you would want to, let's say you have a theory that two proteins. Let's call them protein A and protein B. Protein A and protein B are two subunits of an enzyme, right? So if you want to prove that protein A and protein B are two subunits of an enzyme, you can go part way to proving that by proving that they're in the same place at the same time. And you would do that by doing a co-localization experiment. Okay, so you want to prove that proteins A and B interact with one another. You would do this by labeling. You would take a slice of a cell, and then you would make antibodies that will stick to protein A, and you label them with fluorescein so that they will appear green. And then you make antibodies to protein B, and you label those antibodies with Texas red so they would appear red. And then what happens if you have red and green fluorescence in the same place? If you mix red and green fluorescent light together, you actually get yellow. So if those two proteins are in the same place at the same time, you will see a yellow spot. If they are in two different places, you'll see red spots here and green spots there. But if the two proteins are occupying the same space, then you will see a yellow spot. Okay, so you make antibodies to protein A, you label them with Texas red. You make antibodies to protein B, you label them with fluorescein green. You put the antibodies onto the slide at the same time. When red and green light combine together, you get yellow light. And so this is what you see in a co-localization experiment. Right? So these are actually eggs from a fruit fly. These are Drosophila melanogaster eggs. And somebody is trying to prove that two proteins co-localize to the same place. And so if you look at the top left, you see that one protein called WOR, W-O-R, is labeled in green. It's labeled with antibodies in green. And then next to it, there's another protein called delta that's labeled in red. And then if you combine the two images, you can see all these yellow spots co-localizing along what is about to become the Drosophila, the fruit fly's spinal cord. And now, fruit flies don't have vertebra; they are invertebrates, so they don't have a backbone, but they do have a band of neurons that kind of forms a rudimentary or an elementary spinal column. And so these scientists were trying to prove that the proteins WOR and the protein WOR and the protein delta work together to cause the development of the Drosophila spinal column. So that's a perfect example of a co-localization experiment. Okay, here are other examples of co-localization experiments. You label one protein with a red antibody and another with a green, and then when you see them together, as you see on the bottom right, then you see a yellow spot. All right, so this is to summarize the different staining techniques that we've gone through. We talked about general versus oversight stains. We gave several examples. We talked about stains that we use with bright field microscopy versus stains that we use with fluorescent microscopy versus stains we use for electron microscopy. We learned about differential staining methods. The most obvious one, the most basic differential staining method you have to learn is the gram staining protocol, including the uses of crystal violet, iodine, alcohol, and safranin. And you need to know the other stains that we use to identify specific types of bacteria, including the carbylfusion, and which stains mycolic acid, which is a way of diagnosing whether or not somebody is infected with tuberculosis, or you're staining with malachite green to find endospores to see if somebody is infected with anthrax or other things like that. Okay, so you might want to try this exercise. Can you take all of these stains that are listed on the left and match them to all of these techniques that are listed on the right? See if you can do that, because that might be one of the tests that I ask you about. That'll be probably an exercise like that will appear fairly soon in the weekly quizzes. Okay, so in this very long lecture, I apologize for it being so long, we reviewed the basic structure of cells. We discussed some of the types of organisms that we study as microbiologists. We discussed which types of microscopes we use to study them, and we discussed the types of stains that we use to visualize them under the microscope, and those visualization techniques are often used for diagnosis. So if somebody is infected with a bacteria or a yeast or something like that, we want to know which one it is so that we can select an appropriate drug or antibiotic to kill it. right? Because all antibiotics do not work on all types of bacteria. For example, you need to use one antibiotic to kill one type of bacteria and another antibiotic to kill another. And so we need to know what you're infected with before we can select an antibiotic to cure you. We will discuss this technique of selecting antibodies quite a bit later in the course when we discuss the specific mechanisms by which antibiotics kill bacteria. But for now, just know that it helps if you can diagnose which bacteria somebody is infected with. That will help you to pick the appropriate antibiotic. Okay, thank you very much. And now you can proceed to Unit 3, which is a discussion of ways in which bacteria and yeast and viruses make you sick, which is known as pathology, and the way that we study the distribution of illnesses over populations, which is a branch of science called epidemiology.